Everyone, I would request to kindly keep your phone on silent mode and take your respective seat. Good morning, ladies and gentlemen, esteemed guests and conference participants. Welcome to the second day of our fourth National Conference, Health Margdarshan 2025. The main theme is Unlocking Potential, Academia and Industry Partnership for Transformative Healthcare Research. My name is Ananya Chanda, Assistant Professor from the Department of Pharmaceutical Technology, School of Health and Medical Sciences, Adams University, and it is my pleasure to be your host for today's event. On behalf of the university and the organizing committee, I am delighted to welcome you all. We are so pleased to see you all here, both in person and those joining us online, to engage in meaningful dialogue on the topic of unlocking potential, academia, industry, partnership for transformative healthcare research. This conference is a vital opportunity for us to exchange ideas, showcase groundbreaking research, and foster new collaboration that will shape our future. We are honored to have such a diverse audience of students, faculty members, researchers, industry professionals, and chief dignitaries joining us for this important discussion. The primary goal of this conference is to explore the dynamic partnership between academic pharmacy programs and the pharmaceutical industry. This collaboration is not only essential for scientific advancement, but also for ensuring that future pharmaceutical technologists are equipped with the knowledge, skills, and practical experience necessary to meet the ever-changing healthcare landscape. Collaboration between pharmacy education and the pharmaceutical industry plays a crucial role in advancing healthcare innovation and improving patients' outcome. By working together, academic institutions provide a strong foundation in pharmaceutical sciences and clinical practice, while industry partners offer real-world insights, cutting-edge technologies, and opportunities for research and development. This partnership fulfills drug discovery, formulation, and regulatory compliance. It also enhances experiential learning for pharmacy students through internship, industry projects, and collaborative research, preparing them to meet the evolving demands of the healthcare sector. Ultimately, this synergy fosters innovation, accelerates the development of safe and effective medications, and ensure a well-trained workforce for the future of pharmacy. With this, we shall proceed with our second day of our conference. It is my great honor and privilege to welcome our speaker, Dr. Saptoshi Chatterjee, Associate Director, Incubation Industry Academia, Collaboration Lead, Chief Coordinator and PI, EUVA Center, Adamas University, supported by BIRAC. To today's conference, Dr. Chatterjee is an accomplished academician and researcher whose remarkable contributions in the field of microbiology and biotechnology have set a benchmark for excellence. Dr. Chatterjee holds a PhD in microbiology from the University of Kolani, along with a master's and bachelor's degree in microbiology from Bangalore University. His expertise covers several cutting-edge research areas including nanobiotechnology, anti-cancer drug development and screening, and environmental microbiology. He has made significant contribution in the application of nanoparticles in biological sciences and in the design and screening of anti-cancer drugs, areas of immense importance for the modern healthcare. His academic journey is equally impressive as a postdoctoral fellow at ISAR Kolkata under MHRD, Government of India. Dr. Chatterjee's research profile includes 23 publications in Scopus indexed international journals and 4 book chapters, along with supervising numerous postgraduate and doctoral research projects. His excellence has been recognized through several prestigious awards, including the Albert Nelson Mercuse Lifetime Achievement Award, the Best Leader Award at the Adamas Dialogue, and the International Travel Scheme Award by the Department of Science and Technology, Government of India. He has also been honored at multiple international conferences for his outstanding research presentations. I would request all honored dignitaries, faculty members, participants, and students, please join me in extending a warm welcome and big round of applause to Dr. Saptoshi Chatterjee as we invite him to the stage for his key address and share his insights for this session. Please, sir.
A very good morning to everyone. Good morning, everyone. Good morning. Not loud enough? Good morning, everyone. Good morning. Better. So, it's always a challenge to be the first speaker of the day because you will be eagerly waiting for speakers who have come for listening to the conference. So, I would just try to set the tone of the day to something which probably is sometimes overlooked in the academia and research, and that's the domain of innovation and entrepreneurship. To start with, is there anyone in the audience who doesn't have a problem? Any hands raised that I don't have a problem? So let me consider that every one of us has problem. And why a problem is a problem? Because we don't have solution to it. So, who is going to find the solution? Now, if I try to relate innovation and entrepreneurship or our conventional academic research to the dignity of our individual and in line with the conference of pharmaceutical technology or pharmacy, then I would like to enlist a few names. I would like to speak about a few names and events or innovations that have changed the world and that have emerged from this place, and if not Bengal, I would say it's India. The first is a treatment of visceral leishmaniasis. I know that Arijit sir is one of the speakers today, so I won't go much in depth with visceral leishmaniasis, but what I might say that most of you perhaps have not known the name of Dr. Upendranath Brahmachari, who was the first to use urea silvestamine for the treatment of Kala-Azar, which is known as the drug, a wonder drug. Perhaps we have also forgotten the name of Hemendranath Chatterjee. Again, a name which is not much popular with, but was the first to show today's ORS, oral rehydrating solution, and sometimes it's called oral rehydrating saline, has been first created by him for the treatment of diarrhea, which has now been adopted by WHO throughout the world, and it's again a miracle. The third, which I would like to mention, is a oral contraceptive which is not steroid in nature, and the name is Saheli. Again, an innovation that has came from CDRI Lucknow, Central Drug Research Institute Lucknow. And at the next level, I would like to mention the name of Dr. Subhash Mukhopadhyay, who has given birth through in vitro fertilization, the first in the country and the second in the world, but whose method is still followed in the entire world, gave birth to the lady, Ms. Agarwal, who is known as Durga. Now, all these examples are not a very recent ones. And if you even do a Google to find out what we have been doing in the last 10, 20, 50, 30 or 50 years, perhaps you will not get that impressive count as we spoke or as our history celebrates. Now, what's the reason? I always try to take the role of a customer. I always try to take the role of the affected when I judge something. And in judging the modern pharmaceutical education and research, I'm not speaking about the industry, I'm speaking about modern pharmaceutical education and research, I would only consider successful when I go to a pharmacy shop and I can show, see, this molecule is being used, is in the market, which has been created by me, by my seniors, by my teachers, or from our lab. But do you get that answer? Perhaps not. So even if you go to any pharmacy store, rarely you can relate yourself to any of the medicine which is available in the market. And therefore, if I am ill, if I am affected, I require a treatment, I require a medicine. In most of the cases, the research that we do is important, is significant, but cannot help me as a patient. That's the gap which I feel. The second gap which I would like to talk about is the challenges that we have in front of us. How many of you have a confusion taking a paracetamol 500 and paracetamol 650? Which one to take? Perhaps you are not confused because you have studied a lot about those. But believe me, layman, and the way I say layman is not uneducated man. People who are maybe a PhD in history, engineer and tech, he himself or he or she is a layman in terms of pharmacy, but still is confused which one to take. So can we go for a personalized medicine? Can we go for, I got a very beautiful idea from a student who said, sir, we are doing a 3D printing of items. Can we 3D print medicine? And the exact amount of medicine which that individual requires, can it come out so that I don't unnecessarily use medicine? I reduce the cost and I use exactly what I require. So what is this different from from the normal or conventional education or research that we are doing in pharmacy. And by the word pharmacy, I do not only mean the B.Pharm, M.Pharm people what they are doing. It's entire life science, health and other domain together in an integrated way. India has shown the world during COVID that during crisis, we can do things at a very lightning speed. But is our curriculum equipped to do that? Are we equipped to do that? Or at all, are we having that intention? Everywhere, yesterday being on the dais, it was so wonderful to listen to the industry-academia gap. I have a very personal thought on that. The industry-academia gap, when we were students, we listened. You are listening now, and when even someday you come to the stage, you will be listening the same. So how do we bridge it? There is a complete misfaith because academicians think we are having a superior degree, we are creating the mankind, we are training them, so we don't require industries, only excluding for the requirement of a placement. And the industry feels that we are quite ahead of it, quite ahead of academia. So whatever comes out of the academia is not suiting us. Now let me play the role of the devil's advocate, and I always advocate for the academia, saying that if you go to the traditional innovations in the entire world, you will see most of the innovations have not emerged from the industry but from the academia. And then it was translated in the industry. Be it the Google that we use by Larry Page, it emerged from the algorithm which he created during his PhD. So most of us even need to start thinking that academia should be ahead of industry, and it's not a bridging the gap between industry and academia. It is how we can take academia beyond industry so that whatever we do today, industry will adopt it tomorrow. Then in doing that, if we consider for the time's sake that academia is behind industry, then how do we pull? I was listening to a wonderful talk. I met with Dr. Mashelkar, who was the Director General of CSIR, and he said, we cannot just take a leap, we need to take a frog jump. A frog jump means we have to take a double leap. One which will make us equal to industry, and the second one which will make us ahead of industry, and then only academia will survive. And why I say that? Today a lot of bedside devices are available. Are we encountering the correct problem? See, the problems are very simple in the world. All of us have problem, a universal truth, and every problem of the world can be categorized into 4 types. It's a personal problem, technological or scientific problem, business or financial problem, and the 4th one being social problem. So we may think that this is a personal problem, so who will try to solve it? Believe me, there are millions of people having the same personal problem. So in academia, we often thought that the end product of our research is a research publication. And that's the reason perhaps why we kept on going backward, or industry kept on moving ahead of us. A research publication, a patent, all of these shall be considered as a byproduct of our research. While the product is when you see the people getting benefit out of it. I do not ignore the necessity of basic science. I do not ignore the necessity of the science that you do in the laboratory. But believe me, in this world, if you are not fascinated of converting your idea, your research into a product so that it goes to the market, and if you start feeling that someone else will do it for you, we are fooled. So the same people going to an industry is creating drug, is doing clinical trial, is going ahead in production and selling it in the market, earning money, while we still are stuck in the research. And that gap, what we call, or what the government is now focusing, is called the translational research, where we need to translate what we have thought translate what we have learned. So the knowledge, skill and research what we do need to be converted into something which will benefit the society. I'll just share a small personal experience with you. When I was doing my postdoc at IISER Kolkata, outside the room, our laboratory, there was a beautiful poster saying Anti-Cancer Drug Development Laboratory, Medicinal Chemistry Laboratory. So one fine evening at around 8 o'clock, the security guard entered our room and said, I will quote it in Bangla so that you understand the gravity of it. Dada tumra cancer er oshud banao. Believe me, we were in cloud nine, we were so proud, yes we do. Few of us, the research scholars, I was a postdoc that time, but there are so many PhD scholars who stopped their work and said, yes we do, we are preparing this, that and all. The next moment, the person said, Amar meter cancer, doctor boleche kono oshud nei. The same pause, the same silence was there in the room. Because we thought that whatever research we are doing, will it ever come to the market? Because until it comes to the market, we are not going to help the person who is suffering that disease. And then we started analyzing. I had a conference just 2 days after that, and I started collecting the number of anti-cancer compound, and I was fascinated to see the literature report of anti-cancer compound. I am not mentioning the word drug. Anti-cancer compound is higher than the number of cancer patients in the world. So anything I prepare, I do a little bit of study, I publish it in the paper, that's good academically. But the day we start saying that until it goes to the market, until the problem you are trying to address is solved, you may not have the capacity to solve it for billions, but even for a single person, until it is solved, until you are passionate enough to take your baby, your molecule to the next level, industry will always be ahead, and we will always keep on saying industry-academia gap. So the first gap is in our mindset. We need to address them, saying that our research should yield something beneficial to the society. I repeat that I am not ignoring the necessity of basic science. I am not ignoring the necessity of creation of knowledge. But in today's world, which is full of data, again creating some junk data has no value to the community or the society. And as a result, you may see that most of the institutes are now in dearth of funding. Why? Till date, we are always dependent on the government for the funding. Because, again, I asked the same question to one of the very, very senior scientists from DST, and he said, in my last 20 years, I have never seen a faculty, a PI going to the review committee and said, I have failed. Everyone is successful. So we should get rid of this mentality. We should know to fail. We should encourage failure. The greatest fear we have created among students, and even it has gone to researchers also, that the fear of failure. I can understand as class 5 student fears that if you don't study, you will fail in the exam. But if the same fear remains in a research scholar, and he doesn't try something new, he doesn't try to be innovative, if he doesn't try to disrupt the industry and does everything routine, then believe me, what has brought us here was a little bit of knowledge and skill. A very, very insignificant knowledge and skill I do have, and that has brought me to the podium. But believe me, if the next generation, the students sitting in front of me, if you acquire 20% more than what I acquired, you will not get a job. That's the reality. So you have to start thinking something different, start going beyond this, and there is a lot of problems to address. Now pharmacy stands a very, very unique thing, because it comes under the Ministry of Health and Family Welfare, and many a times it also is linked with the Ministry of Chemicals and Fertilizers. So you have the entire world in front of you. We all speak, I will just deviate from pharmacy and go a little bit to the domain of chemical and fertilizer. A couple of years back, we have some agricultural land which is very near to this. And we students of biotechnology, we always learned about biofertilizer. So we went to the field and asked the farmer, don't you use biofertilizer? The farmer was witty. What he said, sir, take a 360-degree loop and tell me which plot is using biofertilizer. So we just turned a 360-degree view and we saw every plant healthy and grown. In one place there was all the plants just like the post-lunch session. Drowsy. Not enough of nutrition or something. I said, is that one? He said, yes, that is the one. So we may glorify things in the textbook, but in the field what is happening we need to understand. And there is a lot of possibility. So I would urge anyone who is studying not to get confined to knowledge, because knowledge will not take you to the next level. That days are gone when we were taught by teachers who are knowledgeable, even if they don't have the skill to teach. But they earned well, they got good position in the world because of the knowledge. We are perhaps the product of those generation where knowledge and skill both were required. But you, the next generation, even if you have knowledge and skill, you will remain unemployed until you can solve the problem. And if you want to solve the problem, or if you can solve the problem, believe me, sky is the limit. So at the EUA center that I had here, we have tried to create an ecosystem. We have tried to give something more to those people who dared to think. And in that, we appreciate failure. We encourage trying. It's not possible for the entire university to keep all the labs open till midnight. But how many of you do an experiment at 8 o'clock in the evening? Wherever you are from, whichever college or university you are from, perhaps not. And believe me, I can show you my mailbox which gets flooded every week saying, sir, we want to work whole night in the lab, can we get access? And we provide that access. So we have to think beyond our curriculum. We have to think beyond what is taught to us. Let us make problem solving our motto. Let us use our knowledge and skill towards that. And I've just tried to enlist a few challenges. We start with digital healthcare. Today, any symptoms that you feel, the first thing people do is go to Google, type that symptom, and right from headache to cancer, it matches with something, and people get scared. So digital healthcare is important. We need to understand the course of disease management. Once in a seminar, I met a doctor from AIMS and he gave a beautiful problem statement. He said, I don't want medicine. Can you suggest the person who has come to the hospital that which doctor he should go and meet? That is not known to 99% of the people. And you know, in government sector, where they visit thousands of patients within a couple of hours, I asked, how do you do? He said, we start diagnostics right from the time the patient enters. Whatever part in the body he is holding, what is his facial expression, that is the way we start our diagnosis. Because after he sits, we have just 2.5 minutes, not more than that, to do the diagnosis. So can you think of solving these issues? Till now, antimicrobial resistance is one of the greatest problems, and perhaps unregulated use of antibiotics is the reason. So, can we take up these challenges right from our college days? Can we take up these challenges during our PhD? Because these will solve the pertinent problems of the world. And I started with problem. And let me tell you, the main reason why I like to interact or address people is not because I know something. People know better than me. But I have one understanding. A problem is a problem because we don't have a solution. So my grey matter is incapable of solving, but the youth sitting in front of me really has the capacity to solve the problem because they can think out of the box. because they can think differently. And if you can do it, I am sure that you will have a better world to live in, a better world to keep yourself for. Thank you very much for your time.
Thank you, sir. We are privileged to have him share his insights on our national conference theme. And I'm sure his vision and experience will undoubtedly inspire us all and provide valuable perspectives on bringing the gap between academia and industry for the betterment of healthcare and society. I now request Dr. Sheikh Murtaza to present a token of gratitude and to kindly felicitate our esteemed speaker, Dr. Shoptoshi Chatterjee. I request the sirs to kindly take their respective seats in their designated area outside the stage. Thank you. It gives me immense pleasure to be here among such distinguished academicians, researchers and industry experts. With that, let us now move forward with our scientific sessions. We now begin scientific session 4. I request our respected chairperson, Dr. Arijit Bhattacharjee, Associate Professor and Head of the Department of Microbiology at the School of Life Science and Biotechnology, Adams University, Kolkata, along with our distinguished resource person, Dr. Utpal Nondi, Associate Professor, Department of Chemical Sciences, Unified Academic Campus, Bose Institute, to kindly come up on stage and take their seats. To preside over this session, we are honored to have Dr. Arijit Bhattacharjee, is an accomplished microbiologist currently serving as Associate Professor and Head of the Department of Microbiology at the School of Life Science and Biotechnology at Adams University, Kolkata. He earned his PhD in microbiology from the CSIR, Indian Institute of Chemical Biology, IICB, Kolkata. Following an MSc in microbiology from the University of Calcutta, Dr. Bhattacharjee has over a decade of academic and research experience, including roles as postdoctoral researcher at the University of Lowell, assistant professor at Tripura Central University, and assistant professor at Presidency College, Kolkata. He also served as CSIR Junior and Senior Research Fellow at IICB. His research focuses on antimicrobial resistance and resistomics, molecular parasitology, microbial genetics, and biofilm cells. He explores innovative strategies such as quorum sensing regulated bacterial metabolites as alternatives to antibiotics, studies macrophage modulation during biofilm infections, develops molecular diagnostics for resistant infection, and investigates molecular evolution and drug resistance mechanisms in pathogens. Dr. Bhattacharjee has published over 20 research articles in international peer-reviewed journals, authored 3 book chapters, and serves as reviewer for prestigious journals including PLOS, Neglected Tropical Diseases, and Frontiers in Pharmacology. May I now request Dr. Arijit Bhattacharjee to kindly introduce our esteemed resource person, Dr. Utpal Nondi, and would request to extend up to question-answer segment of this session. Sir, please.
Good morning to all of you. First of all, I would like to thank the organizers for providing me the opportunity to welcome Dr. Utpal Nundi, who is an accomplished researcher in the field of natural products in drug discovery. So, Dr. Nundi completed his B.Pharm and M.Pharm from Jadavpur University, followed by his PhD. And he served different eminent institutes, including IPC and CSIR IIM Jammu. Presently, he is working as associate professor in Bose Institute. So he has a decade-long research career, actually more than a decade-long research career, with 100+ publications, and several PhD scholars worked under his supervision. And his research endeavor actually culminated into several phytopharmaceutical drug development. He is fellow of Indian Chemical Society as well as of MASI, Royal Society of Chemistry, American Chemical Society, and many other eminent research societies. He has received several awards, including several best paper awards and UGC research fellowships. Okay, so let us get enlightened through the glimpse of some of his research in the field of drug-herb interaction and translational pharmacology. The title of today's talk is the same. Is Opportunities and Challenges in Inhibiting Cytochrome P450 Enzyme for Drug Discovery. So let us get enlightened for the next hour. Thank you. Thank you, sir, and welcome. The session is yours.
Thank you very much to the organizers for giving me opportunity to present my little bit of research work as well as what I can perform in my own laboratory, as well as some specific focus on drug discovery field and just briefly, I started my education in Jadavpur University, completed my UG, PG and PhD. Completed my UG, PG and PhD from there. After that, I started my career in Indian pharmaceutical Then I moved to CSI Indian Institute of Integrative Medicine, where I work in the PKPD toxicology division. Basically, my responsibility there on pharmacokinetics and toxicology. I moved to both into basically towards the more and more basic research, including my experience, which I'm going to apply of having the translational research. Basically, if I talk about my research expertise, it will mainly focuses on pharmacokinetics because basically I am dealing with failures. What the previous speaker said about, because pharmacokinetics is one of the steps in the drug discovery process where the molecule fails, having a lot of issues related to viability and other pharmacokinetic parameters. If I segregate into different parts, if I talk about absorption, there are compounds having a lot of solubility issues. Though some formulation strategies are using to solve these issues, but there are some lacuna of the class species class for drugs, which are people generally unable to solve these issues of low solubility that will affect on biology. Others like lipophilicity, drugs have to have optimum lipophilicity so that it can reach better absorb, it will affect on biology. Some ionization constant, obviously, if I talk about oral absorption, because how much extent of action it will affect on absorption behavior of that particular molecule. Chemical stability, we already know some molecule having stability issues in the stomach, so that it will be given in different formulation form, so that it can be absorbed from intestinal form. Several WHO strategies are used to deliver this kind of molecule having some issues. Permeability of density, this is one of the key rate limiting parameter I can say for absorption of a particular molecule, and with fast transporter, if I talk about combination strategy, that matters. If I talk about distribution, some of the product strategy people are using because of plasma stability. Issues can be used in the form of advantage, because some pyramide type of molecule, when it reaches to the plasma, then it will open up to its active molecule. If the active molecule directly given into the oral route, it may be possibility of degradation in the stomach. Same in case of markaptopurine type of molecule which are given in the arythrothyroid form. Plasma protein binding, though it's not the concern, because free fraction is important rather than how much. It's 99% plasma protein bound. This is okay, but free fraction, how much it is available, this is related to efficacy. That should be taken keep in mind with because during the evaluation of this particular parameter, with respect to particular parameter of a compound to be druggable. RBC partitioning, we know protein is type of compound which bind to the RBC and cause some toxicity. Therefore, it is very much important to elucidate this type of parameter during the preclinical drug discovery process to know how the molecule behaves. It will also help which biological matrix have to be focused, because during the in vivo stage we have to choose a biological matrix so that the target analytes concentration will be measured there. Therefore, serum, plasma, whole blood binding,
like tacrolimus type of molecule, it will remain bind to the RBC. That's why whole blood analysis is important rather than plasma and serum. If you go for plasma and serum, there must not be fancy time in the doctor. Tissue distribution, obviously this is one of the parameters you have to, during the in vivo stages, you have to evaluate it, because some of the compounds have the tendency to bind to the particular tissue. Like if I talk about globin type of compound, it may bind to retina and cause some retinal damage type of toxicity. At the same time, if I go for discovery of a anti-TB drug, if it is reached to the brain, it may be useful in other aspect. Because if it is reached to the brain, then it can be used some other purpose where TB infection can go to the brain. In other, my target is not the CNS related TB treatment, then it's an disadvantage. We have to think about the risk-benefit ratio as far as the therapy is concerned, but whatever target we want to achieve. Some of the metabolic, this is the most common problem of any drug molecule when I thought about metabolic stability. Therefore, when any drug discovery process, if any problem is the metabolic stability, first in vitro level will choose different set of microsomes, so how much metabolic stability is. Because if I use some 6 different, because if I choose human liver microsome, mouse liver microsome, rat liver microsome, the data can extrapolate to the how maybe the in vivo behavior, because we can choose some certain type of parameters that can predict the in vivo what type of behavior it will show based on the in vitro data. Because in between there is some, what is the protein content in the liver, how much liver weight, and what is the blood flow. That is the three parameter it will involve to calculate from the in vitro data to in vivo data. Then metabolic identification, obviously, what type of metabolite it will generate, it is active or not, that is one of the important concerns. If it is inactive, then we have to think about this is the major route, obviously. And CYP inhibition, this is basically concerning the drug-drug interaction perspective. And some excretion routine which it will excrete. I have also exposure of toxicity. It may be regulatory toxicity, it may be disease model, because I am interested in disease state kinetics. Normally, when I put the data of any kinetics, evaluate the data of any kinetics, it may not be the same in case of disease condition. Disease to disease may affect the kinetics of behavior of this particular molecule. As well as can develop certain drug-induced liver injury model, cardiac injury model, to elucidate what type of protective toxicity a compound can give. Among all these, my key interest or focus of my research was basically on cytochrome P450 regulation in different disease states. During this tenure in CSR I am involved in several drug discovery processes, as well as involved in some solving the issues of phytochemicals. Basically, my research focused on natural product pharmacology. So I am contributed to some drug discovery process having this type of molecule having some issues. Mainly, if you think about this is the cannabinoid drug discovery process, because this is one of the key molecules for in today's scenario. And if I say, as far as drug discovery concerned, this is the 6-7 molecule under different discovery stage with respect of cancer, with respect of some gastric ulcer, because some are during the IND filing phase, some are going to start the phase. Now come to my topic of my today's talk. What are CYP enzymes? Basically, cytochrome P450 enzymes, we all know. If any doubt or question in between, you can directly ask me. I am happy to answer. Cytochrome P450 basically a superfamily of enzymes, membrane-bound protein. Basically, when I talk about cytochrome P450, we think of the liver. Not only the liver, beyond liver, it will present like gut, and as well as other organ, it will be present, even in the brain. The cytochrome P450 present, because recent research work said that it may be cytochrome may play a role in some transformation of the cholesterol, or you can say cholesterol biosynthesis procedure, so that it will affect on liver. If I differentiate the role of cytochrome P450, it will involve the bioactivation process, several molecule bioactivate to its active form. On the other hand, we already know that this is a drug for drug metabolism. More than 70-80% of the drugs are metabolized by cytochrome P450 by using oxidation process, hydroxylation process, and methylation and other process. Among the cytochrome P450, these are the 7 C's USFDA recommended during the evaluation of the drug-drug interaction in the in vitro level during the drug discovery process. So CYP3A4 contributes largely for the metabolism of major drug, and other than this, CYP1A2, CYP2C9, 2E1, 2A6, 2B6, and 2B6. If I differentiate the drug example, differentiate with an example of how CYP-mediated drug metabolism occurs. If I talk about diclofenac, we already know that this is a NSAID molecule. This is by CYP2C9, it will metabolize by 4-hydroxy metabolite, and CYP3A4, it will convert it to 5-hydroxy metabolite. Simple, I can think about when the study have been done to identify the metabolite. Most of the people use some mass and NMR to differentiate this molecule. If you see these two metabolites, it's very difficult to differentiate what is going on in the metabolic process. And if I think about cholesterol type of molecule, some 46A1 will play one of the major role during the metabolism and biosynthesis process of cholesterol. Right now, that's why people are working on C46A1. If any cholesterol biosynthesis can be intervened in the cholesterol biosynthesis process, so that it will affect some disease. What are the impact of drug interaction? We already know that when I talk about cytochrome P450, drug interaction is highly related to it. Because when one drug having the substrate of particular cytochrome P450, that means metabolized by particular cytochrome P450, another drug which is hampered to that cytochrome P450, it may be in the form of induction or in the form of inhibition. Obviously, the plasma concentration effect. If there is any co-administration of a inducer, obviously there is plasma concentration will be dropped. And in that case, if it reaches to the subtherapeutic concentration, obviously therapeutic efficacy we cannot expect. In that case, if I talk about antibiotics, in that case there is a chance of growing resistance with the co-administration of these two drugs because of the drug interaction. On the other hand, if I talk about cytochrome P450 inhibition, if it is inhibited, obviously metabolism hampered, the plasma concentration will increase. In case of increase, it may enhance, go beyond maximum cell concentration. In that case, there is a chance of toxicity if the drug having the inherent toxicity or dose-dependent toxicity. Therefore, the two things very much correlated with the drug-drug interaction. That is, one is therapeutic failure, and another part is the precipitation of the toxicity. If I talk about pirapitin and rifampicin, this is the two drugs given simultaneously. Because rifampicin earlier used nowadays the online drug for anti-TB therapy. And when I talk about betaquilin, this is also a CYP3A4 substrate, means metabolized by CYP3A4. And rifampicin, this is a strong inducer of CYP3A4. Therefore, when I go for combination, the betaquilin plasma concentration will be dropped. And in that case, we are not getting the anti-TB efficacy which I expect due to betaquilin therapy. In that subsequently may enhance the chances of growing resistance. Right now, both are not used. Right now, if we do this preclinical data, later on, people are not thinking about the combination of this because beta-choline are reserved for that reason, reserved for multi-drug resistance. And another example, fluoxetine or morphine, when I go simultaneously due to strong inhibition by of C2-C by fluoxetine, morphine concentration efficacy will be lost. If I talk about adverse effect, then if I co-administer copidogrel, which is basically blood thinner, you can say, with omeprazole, in that case, due to inhibition of CYP2C9 by omeprazole, copidogrel plasma concentration will be increased. Obviously, the active form, there is active formation of its metabolite will be overall it will affect on efficacy, because copidogrel as such is active, its metabolite is also active. Another example of paroxetine with amitriptyline, which basically enhance the toxic effects because of inhibition of CYP2D6 by paroxetine. Till now, I am talking about only the worst scenario of drug-drug interactions. Is there any positive? Yes, there are positives. If I think about the therapy of anti-HIV therapy, right now one drug is available which is only, there is no anti-HIV efficacy of its own. Provisister is such a molecule, it only inhibits the CYP3A4. That's why it will be used in combination with darunavir alone, as well as combination of lopinavir, afinazepam and darunavir, so that it will increase the or boost the efficacy of that particular combination or single molecule if used. The same thing you people will observe during the COVID times, because ritonavir is one of the known antiviral drug, but it can inhibit the CYP3A4. That's why when lopinavir is prescribed, ritonavir will be given in combination, not only due to antiviral efficacy. This is related to CYP3A4 inhibition to boost up the efficacy of lopinavir. Another molecule like dextromethorphan, when administered with quinidine, there is augmented oral exposure due to intended drug interaction, I can say, because I intentionally involved this particular co-administration to boost the efficacy. Another example in recent scenario, you can see like CYP17A inhibitor. This is basically abated acetate is a drug approved. This basically inhibits the CYP3A4 enzyme, which basically inhibits androgen biosynthesis procedure. That's why this is prescribed right now prescribing in prostate cancer therapy. If we look beyond this approved drug, if I want to see the clinical pipeline, you can see there are a lot of clinical trials are ongoing because of CYP3, using some CYP3 inhibitor. Like Ovisistat will include, along with paclitaxel, are undergoing clinical trial to check whether there will be benefit or not. In the same scenario, cannabinol as a CYP26 inhibitor use some retinoic acid for cancer therapy, because retinoic acid is one of the endogenous compounds which is involved or dysregulated during the cancer. Overall, the motto of this particular combination therapy using some CYP4 inhibitor is to boost the efficacy. If I look into the how I am going to evaluate, if I am in the preclinical stages, what type of USFDA recommendation? USFDA suggests that this is the index reaction you have to use if you go for any new compound to identify this is a CYP3 inhibitor or not, in the in vitro platform. And these are the inhibitors you have to use as a standard. Other than this, second line inhibitor is also suggested most of the cases. Like CYP1A2, phenacetin 1A2, phenacetin converted to paracetamol by CYP1A2. This is the index reaction you have to use. That means you have to use the reaction by adding the substrate of phenacetin and measuring the metabolite as a paracetamol. Like in case of CYP2D6, bupropion hydroxylation process, in case of amodiaquine, N-dimethylate desmethyl amodiaquine, the metabolite which are going to measure generate by 2C2C. If I talk about CYP2C9, then dihydroxylation is the index reaction used. Yes, methionine, then 4-hydroxylation, CYP2D6, dextromethorphan is the subject to convert, which are converted to testosterone, which is the metabolite. If I talk about CYP3A4, then testosterone 6-beta-hydroxylation are used. That means 6-beta-hydroxy metabolite generation are measured or monitored. Methylene hydroxylation process is also recommended. This is basically for CYP3A5. If I want to define CYP3A4 and 5, then methylene hydroxylation I have to consider. Should be the experimental setup. If I want to evaluate what type of, this is the minimum thing I am telling about, because this is the 3 platform people are generally using, the in silico platform. I can check what type of interaction, molecular docking and other, if I write some other 3D QSR, 2D QSR, I can use some model where I can evaluate what type of protein interaction, what is the extent of interaction, what type of interaction, it will be useful or not. Only interaction I can judge. It may be related to induction or it may be related to inhibition. Then in vitro setup, most of the people are using human liver microbiome to assess what type of inhibition using the investigation I mentioned earlier. Then in the preclinical model, you can evaluate what type of drug interaction. There may be a lot of questions regarding the translation of the preclinical to clinical. Most of the things happen if you choose the right species. Because if I talk about mice, rat, dog, monkey, there are a lot of changes in the cytokine behavior. If I close species I am using, if that means I am using monkey, this is good. But that ability should be there. So generally in our lab, we are generally use some mice and rat model, depending on the study. In the clinical setup, this is, you people know, this is risky and as well as costly. There is no drug-drug interaction behavior are going to evaluate with respect of by doing clinical trials. Obviously, if any safety concern is, there is a question, then appropriate clinical trial are generally planned and executed. As a whole, if I talk about a candidate, pinosemine, we observe that this is a potent inhibitor of CYP1A2. How? The same procedure, we applied some in vitro in silico strategy, applied some in vitro behavior, which type of, how much the safety, which category it is. Because if I talk about in vitro cytokine inhibition potency, then 1 to 10 micromolar, I can say this is a moderate to potent, strong inhibitor. Above 10 micromolar, if it's the IC50, we can say this is a weak or no inhibition. And below 1 micromolar and onwards, we can say potent inhibitor. Below 0.1, this is a highly potent inhibitor. But this is the general category. If the plasma concentration of a particular drug or molecule reaches to the particular organ or blood, higher than the IC50, you can expect that inhibition, obviously. But depending on the mechanism of inhibition, the time span of concentration it will remain, that may be one of the correlation you have to draw. Where we observe that this shows some IC50 of below 1 micromolar, having some mechanism of inhibition type of mixed type of inhibition following some competitive behavior. We saw a lot of interaction with the human CYP1A2 enzyme and other whatever. When you do the simple experiment, you using microsome, you have to have sure that this compound will not break in the microsome. This is the small experiment we have to take in care, because if it is decayed, how can I assess that this is going to inhibit or not? If there is an inhibition and compound will break into the liver microsome, then evaluation procedure there is wrong or faulty, I can say. At the same time, you have to have, when you go for in vivo model, you have to have particular blood or tissue. If you look for some tissue, you have to have measured the in simultaneous experiment. From simultaneous experiment, you have to obtain that biological matrix in parallel and check what the concentration of the target allele is. Not only the observation that this is the difference in plasma concentration, because caffeine is a substrate of CYP3A4, so CYP1A2. This is all about drug-drug interaction. That means we can conclude that If I take pinoceramine and this much concentration, if it is reached, there may be drug interaction. Drug means nutraceutical drug or food drug interaction you can see, because pinoceramine is present some of the honey type of compounds. Probably. Now you see the other part. There is recent evidence of lung cancer at C1 level is upregulated, highly upregulated, because some nicotine-derived molecules generally activate the C1 at higher the level. If you think about this type of C1, then you can target this type of inhibitor where lung cancer highly upregulated C1 is there may be chances of efficacy. Till now, I am talking about the drug-drug interaction or some drug, that means foreign particle. If I think about what is the status of endogenous metabolism, a lot of things are reported in recent times which are involved or disregulated, you can say, in the different disease states. You can say C2A is one of the cytochrome P450 we already know from 20 years back. But if you see the literature, right now this literature is coming on C2 on 2A6, this is involved in cancer pathogenesis. These are the different disease states, I can say, where the CYP are dysregulated. If I talk about NFLD, high level of CYP is there. If I talk about diabetes, lower level of CYP. What we can conclude from this type of information? For example, if I am going to treat a diabetic patient, CYP level is low, that means if I, during the PK study, if I check in the normal mice or rat, we can get some observation. If I induce diabetes, then I check different observation may obtain. You have to, there is requirement of dose adjustment because of CYP behavior. Because if I talk about sulfonylurea, this is metabolized, most of the sulfonylurea are metabolized by CYP. Therefore, you have to check this type of information before the dose regimen, fixing the dose regimen as well. Therefore, disease state kinetics is very much required to know before the further progress to the efficacy study during the drug study or preclinical stages. If I talk about cancer, there are a lot of cancer cases where clinical data suggests that they check in parallel to, if I talk about breast cancer, they check the cancerous tissue in comparison to the non-cancerous adjacent tissue. What is the upregulation, downregulation cases? Most of the cases, these are upregulated. Some endogenous cytochrome P450 enzyme which are involved in the endogenous metabolism, right? C2A2, one of the C2A2, one of the cytochrome P450 which is involved in the metabolism of arachidonic acid. Therefore, one of the key area of this is the arachidonic acid metabolism pathway. You can say the C-picoxygenase pathway. My recent time the study, most of the study focuses on this pathway. What happened? Phospholipase A2 from membrane-bound lipid through phospholipase A2 arachidonic acid generated. This arachidonic acid converted by COX to prostaglandins and thromboxanes. We know the NSAIDs are the drug which inhibit this stage, and there are a lot of drugs in the market based on this conversion, by blocking this conversion. On the other hand, LOX is another enzyme which is responsible to generate the leukotrienes. By inhibiting using some leukotriene antagonists, there is drug, molecular type of drug, which are available in the market by blocking the LOX enzyme. Another pathway which is AGT, this is minor pathway, minor role of this pathway for the metabolism of arachidonic acid. If I think about arachidonic acid to EETs, EET is epoxy-eposulfite acid. This is the basically having the pleiotropic biological activity. If I think about cancer, this is a tumorigenesis molecule. it will self-proliferate, it cause the proliferation of cell, it cause the increase the motility, induce the apoptosis. Therefore, in the cancer, if there is some research done by some group in the US, they observe that CYP2J2 is upregulated. Therefore, there is a chance of higher EETs. Therefore, chances of more treatment diseases. So my hypothesis is, can I block CYP2J2 so that EETs level can be reduced, and there may be chance of benefits. But it's not because human or any system is very smart in that respect. There may be chance of, other than arachidonic acid, this is the main prevalent KUPA. Linolenic acid pathway is there, ecosanoic acid pathway is there, DHA is there. That can convert it to the subsequent metabolite and cause some effect. Other than arachidonic acid, not much known till date on what is contribution towards the cancer. Then EET is converted to DHET. This is basically the EET is I can say 90% molecule, DHT is the pro-inflammatory molecule. DHT means nothing useful. This is done by soluble epoxyhydrogen. Some anti-Alzheimer work is going on in the clinical levels by inhibiting soluble epoxyhydrogenase so that EETs level can be up. In that case, EETs level lower is available. If I talk about what are the data available, you see that EETs increase the tumor cell proliferation, inhibit apoptosis, promote the cancer cell metastasis. These are the information available. And if I talk about what are the data available, only in vitro 4-5 in vitro data and one in vivo data is available where some clue is there to epoxygenase inhibitor or specifically CYP2J2 inhibitor or EET inhibitor. This is the three chances. Can work. So therefore, my target is to elucidate there are 4 regioisomer. Which regioisomer is prominent or most active among these 4 regioisomers? Then, if I inhibit the CYP2J2 and can able to lower the EET's formation, it may be useful or not, can be established as a target or not. Because EET through which pathway it will act, the signaling pathway is non till date. Only little bit information on AKT activation is available. And obviously the target is to, can be a new target can be identified through this pathway, inhibiting this group or this is my ultimate goal. Then the series of assays you have to establish, starting from, if I talk about, I want to look some scaffold to identify this scaffold can inhibit the CYP2J2. So that later on, if there is any lacuna, we can change or design biosynthetic molecules. And use some human liver microsome based strategy to elucidate what type of at in vitro level, which is the most potent inhibitor. Later on, you can establish what type of mechanism it will involve to inhibit this cytokine P4. At the same time, at the in vitro level, you can check this as having action to lower the EETs or not. By using substrate arachidonic acid, you can check easily in the microbiome study. Then, in the in vivo level, you have to establish that this will work on the in vivo level. If I think about substrate, particular 6 substrate you have to take. If I talk about substrate, right now only Margdarshan is the substrate people are using, not more than, no any other substrate is till in the literature. Then validation. I can start from a 31 based model, then go to the Genogam model because Genogam model is more costly and more not accessible to everybody. Then, based on the different biological matrices, we can set up experiments, we can assess what is the condition by Western blot, we can check what is the status of 2J2. By LCMS, we can measure what is the status of EETs and we can correlate. Other than efficacy parameters like tumor volume, tumor weight, and other markers related to apoptosis and agenesis and metastasis. Ultimately, you can use some database as per whatever the US FDA drug available, you can screen for CYP2J2. Why you are going to screen? One can ask. if I talk about tyrosine kinase inhibitors, sorafenib among the all is working very well on the tyrosine kinase. If I go through the cytochrome 450 profile, inhibiting profile, we can see that this is a dual inhibitor of CYP2J2 and 2J2 and 2CA. That may be the one of the reasons, but right now if people are not know the literature, there is no literature that this may work. Have some preliminary data based on this I am told. Our target is to find out a potent inhibitor, is find out a selective inhibitor. Obviously, if it is it should be druggable, so that the preferred or conventional route is the oral. Now, we already observed some behavior of proceeding. That's why I am explaining this behavior with respect of proceeding in our lab. Based on the druggability aspects of proceeding elicited in our lab, we observe we have druggable features definitely with some lacuna. With what are the lacuna? Have some low water solubility of this molecule that can be tackled by formulation approach. Low chemical stability. This is the one of them because this is a single dyne type of compound with respect of light in medical and some PGP induction ability. If I talk about proceeding as a molecule for alignment, then PGP induction may be the advantage because this is going to establish a target because PGP induction is required to out of the brain the amyloid beta from clearing the amyloid beta from out of the brain. If I talk about some cancer therapy perspective, induction, we have here if we go for any combinatorial therapy, there should not be a PGP substrate. In that case, proceeding may induct that particular substrate and cause some hospital-related drug. Different type of substrate I already explained. This is the experimental condition from where we can assess the in vitro. There is the substrate protein and there's some media where you use and some energy substrate like the NADPH and you will incubate for certain specific time in specific condition. Then you use some standard inhibitor in parallel and you can identify what type of inhibition. If I use some inhibition profile for all the panel of seeds by USFDA along with CYP2J2, we observe that most potent inhibition in case of proceeding is CYP2J2. Comparison to other seeds, if I talk about the fold different, lot of fold differences there. That means we can say this is having the selectivity towards the CYP2J2. As per the mechanism of inhibition, this is not a time-dependent inhibitor because time-dependent inhibitor is very dangerous, I can say. If when up to which time we don't like clopidogrel, if I take clopidogrel with some substrate of CYP2C8 substrate like amyloid, in that case this is not inhibiting because but its metabolite having potent inhibitory potency towards the sleep. That means when the to convert it to its metabolite, then metabolite severely affects. There must have near about 20 to 200 fold difference in the potency of this clopidogrel metabolite in comparison to clopidogrel. Other data, which I already explained, that this may be the short protocol from where we can elucidate the CYP2C. It will affect which type of EETs inhibits. It can able to inhibit EETs formation or not from its substrate. And peak interaction finally going to the in vivo. The reversion as a substrate in the presence and absence of proceeding. What is the impact on reversion behavior in the presence of proceeding? We observe that increase in plasma concentration significant fold that establish that this is cause some pressure to inhibition. These are some based on in silico data. We just ensure one type of ensure you can say if you go for screening to selection of a compound, you have to use in the earlier stage. Later on stage we are generally used to before going to preclinical phases because it will give us confidence to use some preclinical model, have some in vitro data and in silico data in our. and it will have some conventional hydrogen bond attract pi alkyl interaction, alkyl interaction is there with the human CYP2A. Now going to the, if I go for efficacy, we use the data, use some 41 induced cancer, breast cancer model, where we observe that there is a lowering of, when in comparison to tumor control, decrease in animal treated with positive, decrease in tumor volume as well as tumor weight. That means it will decrease the tumor volume. Then we check the CYP2A2 expression in the tumor. What is the level of CYP2A2 expression in comparison? So in that case, we check that CYP2A2 expression will decrease in case of with respect to tumor control, where it is elevated. We already have the clinical data. So with respect to non-cancerous tissue, this is anything we observe the similar type of behavior here. Then what is the effect on counterpart? That is what is the level of unities. We also observe that decrease in unity, but it will differentiate with the different unities. If you see, the 11-12 is the most potent percentage of the act on 11-12 is more. Then do some proteomic study to identify what type of protein are upregulated, what type of protein are downregulated, to elucidate what type of overall, based on the KGT database data, what type of pathway generally involved. We also have that this will impact on basically the ECM modulation of this axis. This is all about 2J2, and I can give some glimpse on CYP2E1, another enzyme. I worked on this. 2E1 in normal scenario, this will be
sorry to interrupt you. If you can move a little bit faster, that will be
this CYP2E1, normally there is no enzymatic activity. But when a foreign particle comes, this will start its enzyme activity. And unknowingly it will convert it to a toxic metabolite. Because CYP2A1 not know what CYP2A1 do, because it will convert to A to B to C and other try to eliminate from the body. That is his job, I can say. And unknowingly the compound converted to the B, C and then have some toxic behavior. Obviously, if in a synthetic compound there is a chances of toxicity is there. And due to some free radical generation of the free radical, some toxic effect on the depending on the organ, I can the representative example of liver. Where we observe that we can use different models, animal mouth, rat model, different drug-induced model, and check that CYP2A1 is elevated. But the question remains, today there is a question, it is dependent on substrate or not. Means the metabolite generated have to have the substrate of 2E1 or not, that have to be clarified. Ultimately my goal is to on TB, because most of the TB having some hepatotoxicity behavior is some one or two molecule. That's why finally I want to target how this behavior, patient dropout can be tackled or not because of toxicity, so that it may some aid to patients. We already know the natural products problem. I'm going to the next. If I talk about CYP inhibitor, one of the major issues is the isoform selectivity. Only selective inhibition is very difficult. Those people are trying using some AI as well as some in silico model to develop this molecule. If design can be done, but synthesis of this is challenging. This type of issues with this have some selectivity, isoform selectivity is one of the concern. Other is genetic variability, because see have different allele. Some if there is any impact on particular seed, there may be chances of decreasing of efficacy is there. if I use some CYP inhibitor along with some standard drug, like if I talk about anti-cancer therapy, if any CYP inhibitor along with peptidase it may work, it may give some boosted efficacy. Obviously, additional regulatory requirement is there. I'm just ending. This is my research group, already PhD awarded from I started my journey in CSRT plan, and these are the students already awarded, and the group I left and the group I started here in the research group. Obviously, throughout my journey, lot of funding agencies are there. I have to acknowledge, along with some industry, I must acknowledge and thankful to them for giving me funding to do some research. Thank you very much. I'm happy to answer any question.
Thank you, Dr. Nundi, for your fascinating presentation. And it's really lucid for the students as well as it's deep in terms of application and translational possibilities. So we can have few questions from you, but as you know, we are late, so we only will accept 1 or 2 questions, and then you can interact with Sir if you want to discuss it further. You can raise your hand.
Yes. Yes.
A compound that had some disadvantages, is chemically unstable or kind of CYP inhibiting. So how do you handle if your molecule is chemically unstable? That's my question. Thank you.
If it is a chemically
Unstable, then how do you handle that compound for your preclinical species studies as well as in clinical trial also, if it goes to that phase?
yes. I can say about the candidate, one of the molecule I handle, meropenem. Another candidate I can say rifampicin. Both are drug in the market, but if you keep the rifampicin sample for longer time, in particular solvent, even in water, you will see that with time quinone will be there. If you inject the sample time to time in the HPLC event, you will see the rifampicin peak drops and quinone metabolite increase. This is one. Second, same thing with meropenem. This is my experience during the IPC days. These are the 2 examples. If you keep the sample of meropenem, therefore think about simply how we do the validation. That means, because in case of validation, you have to long-term storage, short-term storage are there. You have to use directly in fresh sample. You have to use as per the validation. This is the one I learned.
Thank you, sir. Okay.
And second, if I think about chemical stability, we have to have changed the approaches. Like if I think about SGA, if it is not, it is degraded in the stomach, I have to adopt some formulation strategy to use some oral disintegrating tablet, or you can beyond that it will release in the intestine, like in case of pantoprazole or omeprazole, like that. However, we have to ensure that it has to be the acidic or basic in nature, which type of nature, proportion of absorption, capability of absorption from intestine or not, we have to ensure that. Like PK will guide you, this is going to absorb or not.
Okay, sir. Thank you, sir.
There is one question from her.
Thank you, sir, for such an informative presentation. I would like to know, there are many breast cancer patients who undergo chemotherapy, and at the same time they can also undergo some other natural products, right? So there must be a cross interaction between this quercetin and those other drugs. So how can we ensure the standard care for the protocols of that? Because it can have some interaction, and it may have some influence which may not be desired by the patient. So how that can be done?
In the textbook language, if I say, right now if you go for any therapy, cancer therapy, doctors are prescribing some juices, specific 1-2 juices they are giving. Their target is to intended drug interaction, to increase the efficacy. I have to survive that patient. If any toxicity, then I will, if any toxicity arises, then I will decide risk-benefit ratio accordingly. I will. But during the chemotherapy, 1-2 juices are, most of the cases are given to increase the or boost the efficacy of the standard anti-cancer. This is intended, but intended drug interaction. Though we know that this combination is going to interact with each other, like if you think about grapefruit juice, though it's not interacting in the liver, this will interact in the gut to improve or cause the toxicity of some molecules. Grapefruit juice is such a potent CP. It will cause, if you take co-administered orally with some standard drug, you will have to have some substrate or inhibitor action, then it will interact with the gut.
Thank you for your question. If you have further question, you can directly contact sir. So I think we should move forward with our next session. I would like to congratulate Dr. Nandi for this excellent work and hope we will get something more in future from his group. Thank you very much.
Thank you very much.
Hello.
Thank you, sir, for such an enlightening session. I'm confident that your insights will provide immense value to all of us, bridging knowledge and inspiring future research directions. I now invite our chairperson, Dr. Arijit Bhattacharjee, to present a token of gratitude to our esteemed speaker, Dr. Utpal Nandi. Thank you, sir. Now I request Dr. Shubhendu Kumar Shahu to kindly felicitate our Honorable Chairperson, Dr. Arijit Bhattacharjee. With this, we conclude session 4. I request to all to take your seats on their designated areas. So we now move on the next session, that is scientific session 5. I invite our Honorable Chairperson, Dr. Shubhobroto Mojumdar, Professor and Vice Principal at the Calcutta Institute of Pharmaceutical Technology and Allied Health Sciences, Uluberia, Howrah, West Bengal, along with our esteemed resource person, Dr. Rammohan Bera, Group Lead, DMPK, Drug Discovery, TCG Life Sciences, Kolkata, to kindly join us on stage and take their seats. To chair this session, we are privileged to have Dr. Shubhabrata Majumdar, who is a distinguished academician and administrator in pharmaceutical sciences, currently serving as professor and vice principal at the Calcutta Institute of Pharmaceutical Technology and Allied Health Sciences, Ulberia, Howrah, West Bengal. With over 17 years of teaching experience and 5 years in the pharmaceutical industry, Dr. Majumdar specializes in pharmaceutics with expertise in advanced drug delivery systems, including nanoparticles, liposomal, and brain-targeted formulations. His contributions to higher education and research are widely recognized. He holds a B.Pharm from Nagpur University, M.Pharm in Pharmaceutics from KLES College of Pharmacy, Belgaum, and PhD in Pharmaceutics from Berhampur University. His research contributions are significant, with 28 publications in reputed journals, 4 patents, and successful guidance of 34 M.Pharm and 7 PhD scholars. He has also authored book chapters on nanotechnology and drug delivery, reviewed for international journals, and actively participated in numerous conferences. Dr. Majumdar's excellence has been recognized through various academic achievements, including securing second position in Bangalore University's M.Pharm program. His scientific leadership and dedication continue to advance pharmaceutical education, research, and innovation. May I now request Dr. Majumdar to introduce our resource person, Dr. Rammohan Bera, and would request to extend up to the question-answer segment of this session. Please, sir.
Hello. Thank you, organizing committee members, Department of Pharmaceutical Technology, Adams University as a whole for inviting me in this August gathering, in this beautiful, well-organized seminar. Present session, I'm thankful to share the present session, which will be an excellent deliberation of a well-known drug discovery scientist, Dr. Rammohan Bera. So let me introduce Dr. Bera. Dr. Bera is a group leader, DMPK Drug Discovery, TCG Life Sciences, Kolkata. Dr. Rammohan Bera is an experienced drug metabolism and pharmacokinetic scientist with over a decade of experience in pharmaceutical research. He earned his PhD in pharmacology in the year 2017 from the Jadavpur University, focusing on the pharmacokinetic and drug-like properties of natural compound, followed by M.Pharm in the year 2006 and B.Pharm 2004 from Jadavpur itself. He worked in leading CROs, pharma industries, including TCG Life Science, Aragen Life Science, Dr. Reddy's Laboratories, etc., where he had led a team of scientists in executing drug discovery and non-discovery projects, supporting global drug discovery pipeline. His expertise spans in vivo, in vitro ADME, in vivo pharmacokinetic in rodent and non-rodent, cytochrome P450 inhibition studies, LCMS, bioanalytical method developments, and so on. Dr. Bera has 18 research publications, contributed several book chapters, and presented numerous number of international conferences. His leadership combines scientific innovations with the operational efficiency, ensuring reliable bioanalytical outcome that accelerates the drug discovery. With his ample of experience, I welcome Dr. Bera with his presentation and deliberation. Dr. Bera, please.
Thank you, sir. So, I think it's afternoon. So, good afternoon, everybody. Am I audible? Last chair. Thank you. So, before jumping into the presentation, so I would like to express my sincere thanks to Adams University and its distinguished professor in pharmaceutical department, as well as definitely the young talent. And it's my pleasure to be here with such type of young talent and curious minds. So before jumping into the session, I just want to, I would like to say that the topic of my presentation is integrated industrial approach for evaluation of new chemical entity. Means whenever a drug, it's a drug after definitely coming into the market. So whenever a molecule is synthesized in the laboratory, that basically in drug discovery setup we used to say it's a new chemical entity because it doesn't have any identity as a drug molecule. It's a new chemical entity like a child born in this world kind of. So lot of rigorous experiments and experimental setup we need to perform for those new chemical entities, and then after that it comes to the market. So before coming to the market, a lot of experiments, lot of animal studies we need to do. I have some expertise in that domain, means in drug discovery domain, particular field in DMPK, drug metabolism and pharmacokinetics. So now it's not working. It's okay. Thank you. Next. Sure. Thank you. So, if we think about the drug discovery process, what does it mean actually? Because as a pharmacy professionals, we are part of the drug discovery process in this world. I would say in global scenario. In not only the discovery process, we are also part of different pharmaceutical industries like production, QA, QC, a lot of things. As a pharmacy professionals, we are there. So if you think about the drug discovery phases of drug discovery, if we divide these drug discovery phases into 6 segments, one is for basic research that first speaker, that is Dr. Chatterjee, has said that is in between industry and academic profession. So I am always with the industry as well as with the academics, because basic research actually comes from the academics. So most of the targeting molecules as well as your target, means a protein or enzyme, whatever is a target for a particular molecule, that basically basic research comes from the academics. So industry just explore those setups as well as those molecules, and under development that molecule finally comes to the market. So if you think about the different segments, one is basic research that takes around 2 to 3 years. There is non-clinical studies that is around 3 to 5 years. Actually, that is trademark. I am working in that field in non-clinical studies. And then it's clinical that is around 3 to 7 years. Then it's new drug applications, NDA. Then it's new drug approval or launch. Then post-marketing surveillance. These are the 6 segments of a drug discovery process. So, if we think about the basic research, what we do in basic research basically we try to find out different type of enzyme, receptor, ligands, targets that we can that is there in the in vivo system, in our body, and that we need to hit by some chemical agent, that may be ligand, that may be chemical entity, so that we can initiate the activity. So that is the basic research part. There mostly we do the synthesis of chemicals, exploration of targets, screening of new chemical entities, formulation screening, and then the non-clinical studies that takes 3 to 5 years. And in that phase, what we do mostly that pharmacokinetic studies in animals, as well as some in vitro studies, some efficacy study, that is means pharmacodynamic, whether the compound is active or not, therapeutically it is active or it will give any activity or not, that we do here, and also some chemical toxicity studies also in that phase. Then it's clinical studies, that is around, if we divide it into 3 segments, that is phase 1, phase 2 and phase 3. As a pharmacy professional, almost accustomed with this process, the drug discovery process. In phase 1, 2 and 3, in phase 1 basically what we do on a molecule, established its a shape and efficacious, then it goes to the clinical trial. And in first phase, what we do, in first phase we test that molecule for a particular healthy volunteer, 5 to 10 healthy volunteer in a human. Then it moves to the patient, whether it is really efficacious or it has the safety as well as efficacy, and then it moves to the phase 3, where actually we do trial for a large number of patients. There also we establish the efficacy as well as safety and any long-term toxicity that we do in post-marketing surveillance. So now if you think about the early discovery phase, means in early discovery phase, what we do, we synthesize the new chemical entity in laboratories, that is around 1 lakhs to 10 lakhs. If you think about the 10 to the 5 to 10 to the 6 compounds, we just synthesize in the laboratory in the early phase of drug discovery. And from that, it goes to the preclinical phase, that hit to lead means the number of molecules that is hitting to your target. That target may be a receptor, that target may be a protein, that target may be an enzyme. So that you can imagine that 10 to the 5 from 10 to the 6 to now it comes down to 10 to the 3. It's very less. Then from 10 to the 3 to lead optimization, it becomes 10 to 100. Then in clinical phase it goes to less than 10, and finally one molecule comes as a drug or as a medicine into the clinical trial. So all those, why 10 to the power 5 to 1 or 10 to the power 6 to 1? So why it is so much of attrition? So much of molecule initially synthesizing and then it comes only 1 or 2 molecules as a medicines in the market. So lot of parameters are there. So pharmacokinetic is one of the most important factor for this attrition from 10 to the power 6 to 1 or 2 compounds in the final stage. So what pharmacokinetic studies actually do, it mostly do that what is the route of administration that is decided in preclinical phase. So whenever we take a medicine, we just take the tablet or capsule through oral route. Who has decided it? That molecule should be taken through oral route or it should be taken through intravenous route. That is decided in the preclinical phase. So that, and at the same time, it is also decided how often you should to take, how much the frequency, whether you will be taking 2 medicines, means 2 tablets in a day or 3 tablets in a day. Who has decided that? That pharmacokinetic study that has been decided. The frequency of dosing. At the same time, whether the molecule will be eliminated from your body, how fast it will be eliminated, or it will be distributed within the body, how fast it will be distributed, or how fast it will reach to your site of action. So all these parameters are shared in pharmacokinetic studies. And what is the maximum concentration where you can expect maximum efficacy of the molecule? So these all parameters are decided in pharmacokinetic study. So I told you that attrition rate. Now if you think about the attrition rate here, attrition rate, you just compare that there in the pie chart you can see that pharmacokinetic parameters, means 4 pharmacokinetic properties of a particular compound that deals almost 50% of the molecule that fails in the trial. So So you can imagine that how much importance it has in pharmacokinetic properties of a molecule in drug discovery. So late 1990 actually, most 40% of the about 40% of the drug failed in the clinical trial. So now it has been changed to that scenario because now it has become to almost close to 10% because pharmaceutical industry now they have changed the strategy how to evaluate the pharmacokinetic properties of a compound in early phase of discovery. So that later stage number of molecules that fail due to poor pharmacokinetic, that kind of cost burden we should not have. We don't want to bear that much of cost burden. So poor pharmacokinetic properties that deals that poor viability of the molecule. Say if molecule is 2% or 3% bioavailable, that means that molecule very little quantity of drug reaching to the systemic circulation that is available for activity. So poor bioavailability of a compound is a great factor for having poor activity of a molecule. At the same time, whether that molecule is eliminated from the body very fast or how that drug is eliminated from the body. Either it is changing to any other metabolites which may be active or inactive, that's a different thing, but whether it is eliminating from the body or it's retaining in the body, how it is eliminating from the body through urine or through fecal matter or any other route of administration, that also a factor. Then undesirable efficacy or safety. These are the parameters that we need to contribute, think about the failure. Then ADME, Dr. Nandi already had described that ADME kind of, so what are the things we need to consider in ADME? Before doing any animal study, means I would say in vivo study, maybe rat or maybe mice, whatever it is. So before doing some in vivo study, we need to establish some data on based on some ADME experiment, absorption, distribution, metabolism and excretion. So if you think about the absorption, what are the experiments that can mimic your whether that molecule will be absorbed into the systemic circulation or not? Means based on some experimental data that will say whether you will take that medicine will go to the systemic circulation without thinking any concern about the drug, whether tablet, whenever we are taking a tablet, we just think that I am taking the tablet means my fever will, my temperature of the body will come down. So we never think about how it is going to the circulation and what is doing inside the body what is doing with the drug. So all these things we can consider here. So now the physicochemical properties evaluation of a particular molecule. What are the experiments that we need to perform to establish the physicochemical property of a drug? Good physicochemical property of a drug. These are the experiments that kinetic solubility, thermodynamic solubility, chemical stability, ionization constant log P and log D. So all these experiments have some value. If we take a medicines, then the tablet goes to the stomach. The tablet will disintegrate, and after disintegration, in that disintegrating granule, that API is active pharmaceutical ingredient is there, and that molecule will dissolve in the gastrointestinal fluid. Am I right? So that gastrointestinal fluid, the drug has to dissolve there. Then only after dissolving, that dissolved molecule will pass to the GIT membrane. So if we test, if we create a system where we can see the solubility of the drug, that will help you whether that molecule will be dissolved in the gastrointestinal tract or not. So that kind of study you can do in thermodynamic stability. Just I am trying to correlate with the experimental data or setup and that with the in vivo condition. Then if you think about the chemical stability, if a molecule is chemically unstable, then it is bit difficult to handle with that molecule. Then you need to take a lot of precautions, modifications of the molecule. Then log D, after dissolving, whether that molecule will partition to the cell GIT cell. I mean GIT cell actually we assume it as a lipid membrane. So buffer as well as your lipid membrane. So that drug has to partition. If it's partition, means from aqueous layer to organic layer, I would say from GIT fluid to GIT cell membrane. So definitely then that molecule will pass to the GIT membrane. So that is actually physical properties evaluation. Then if we think about the permeability, there are a lot of experiments. So based on those experiments, you can say whether that molecule will pass to the GIT membrane or not. So one thing already well known, you know, I think that is a CACO2 cell-based, it's a cell line. So through CACO2 cell line assay, we can say if a molecule has that good permeability, then we can say that new chemical entity will pass through the GIT membrane and it will ultimately reach the systemic circulation. So there are a lot of cell line based assay, different type of inhibitor as well as different type of substrate, efflux protein. So all has that role in to get a drug into the systemic circulation. Then the drug metabolism, if you think about the metabolism, Dr. Nandi already has described a lot of things about the cytochrome P450. So I am not going detail in that because it's a crucial role. So different type of to set up our metabolism of a compound. What are the experiments that you need to perform in vitro? So what we do a liver microsomal steroid study, means from rat liver, say rat liver we just collect a liver microsome that is enriched with cytochrome P450, different type of UGT, different type of aldehyde oxygenase. So these are the enzymes responsible for metabolism of the drug. So these enzymes are present in the liver microsome as well as intact hepatocyte cell. There also we can put a drug and we can see the metabolism of the drug, how it is getting metabolized and what are the metabolites it's forming from that particular system. And time-dependent drug inhibition, that already Dr. Nandi said that time-dependent inhibition is basically when a drug put into the systemic circulation or maybe in incubation in a liver microsome or hepatocytes, that drug itself metabolized to another metabolite which is a potent inhibitor of cytochrome P450. So that is time-dependent inhibition. Now that it's metabolism, the previous speaker already told that different type of cytochrome P450. So a different type of cytochrome P450 to consider. So C3A4 is the major contributor in of drug metabolism in our body. That is more than 40% of the drug. If you think that 36% of the drug, close to 40% of the drug, what we take as a medicine, that is metabolized by cytochrome P450 3A4. So 3A4 has a crucial role in metabolism of drug as well as CYP2D6, that is around 20% of the drug what we eat, or we eat, not eat, we ingest, around 20% of the drug metabolized by CYP2D6, and then definitely 1A2 and 2C919. So here what we can do here is that during metabolism study, or we can say it's a pharmacokinetic study, these all are in vitro study basically. In different enzymatic system is there in our assay system, and there we do the assay and set up the metabolic clearance of the molecule as well as what are the metabolites it's generating in that incubation system, those we used to generate here. Now if you think about the metabolism, there are four phases, I would say. There are different four phases of metabolism. One is phase 0, then phase 1, 2 and 3. So phase 0 is basically, if we think about our body, that major metabolism organ of our body is liver. We know that. And liver contains lot of cytochrome P450 as well as aldehyde oxidase, UGT enzyme, different type of esterase. So all these enzymes are responsible for metabolism of the drug. So here in first stage of the zero phase of the metabolism is that uptaking of that particular compound from your bloodstream to the hepatocyte cell. So that is the phase 0, and then phase 1 is your hydroxylation. This phase 1 is basically some modifications of the compound and some addition or deletion of the structural features of the compound happens in this phase 1. So here mostly that happens, that is hydroxylation, oxidation, reduction, hydrolysis, and then followed by phase 2, that we know that phase 2 different type of conjugation reactions. And then after phase 2, so what is phase 0? Phase 3? Phase 3 is basically efflux of that metabolite as well or a compound from that cell to the circulation, so that it can be eliminated from the cell, hepatocyte cell. So that is basically phase 3. Now here different type of inhibitors as well as substrate. We know the different type of cytochrome P450 is there within our body, and those different cytochrome P450 responsible for metabolism, as well as there are some compounds that inhibit some cytochrome P450. So these are some inhibitors like methotrexate, fluconazole. These are some inhibitors, and there are some substrate. You can see the substrate of particular chips, and there are some inducer. What inducer do actually? They upregulate the expression of different type of enzyme, so that say if you take an inducer, that will induce or increase the expression of say 3A4. So whenever you are administering 2 or 3 drugs at a time, then if the CYP3A4 is induced by a particular drug, say rifampicin, then your drug which is metabolized by CYP3, that will be metabolism of that particular drug will be accelerated. So here is the metabolite identification. Just I have masked some structural part of that compound. So one of the part is the metabolite profiling of a particular compound, because whenever a compound synthesis in the laboratory, I said that is a new chemical entity. So nothing is known to you. You just know the structure and the molecular formula of the compound. Now you need to establish what are the metabolites are forming from that particular compounds, and what are the special type of modifications happening, as well as what are the soft spot, I would say metabolic soft spot, so that you can modify the structure of the molecule accordingly. So new chemical entity has a structural features, and your modification is happening in a particular position, and as a DMPK scientist, if you can pinpoint that particular position, so this is the position that is modification happening by some enzymes. So then the medicinal chemist who is synthesizing the molecule, he will design the molecule in different ways, so that the final molecule what he will synthesize later stage, that will be metabolically more stable. And sometimes it happens that molecules which is binding with the enzyme, that is responsible for activity, means active part of the molecule, sometimes the metabolic modification is happening at particular site. So in that case, that binding site as well as your metabolic soft spot, both are at the same position. So that is very challenging to deal with such kind of compounds, because if you modify the compounds, your structural feature will change, and that will finally lead to inactive compound. So that is very crucial. One of our projects was like that. So yes, metabolic soft spot as well as enzyme binding site was same. So finally that project was submitted just because of the coinciding of these two, metabolic site as well as your active site which is binding to the enzyme. So here, so those you to establish in the clinical phase only, what are the modification and what are the metabolites it's forming, and that finally already I told you that metabolic soft spot, and then it will define a primary elimination. Means what are the elimination pathways, whether your molecule is eliminated from the body through hydroxylation, or any part of the molecule is just simply ND methylation or ND alkylation, or or demethylation is happening and that's why that molecule is eliminated from the body, that you need to establish in preclinical phase. And one more thing, that is that potentially reactive or toxic metabolites. That is one of the major factor in later stage. If it has been recognized in the later stage of the discovery, then that molecule you need to drop that molecule because it is producing on reactive metabolites that is damaging the liver cell. So in that case, if you have that early warning or alert, your molecule is producing some reactive metabolites that is damaging your irreversibly binding with the proteins of the cells and finally it's damaging the cell or liver cells. So that also you can identify in this stage. I request you, please, if you have any question in between, please stop me and you can clarify if you have any questions, please. And so all these things, what we did here is, those are different type of in vitro assay, in vitro studies as well as in vitro experiments with is the pharmacokinetic properties of a molecule. Now, based on the properties that has been decided in vitro experiments, now you move to in vivo studies. There mostly we do some in vivo experiments with rodents mostly, and in later stage we do non-rodents also like dog, monkey, cynomolgus monkey as well as beagle dog. So before going to larger species, we always try to do initially at the smaller species like mice or rat, sometimes rabbit also. So what are the studies that we do to evaluate the pharmacokinetic properties of a molecule in animal? So regularly standardized basis, I would say standard is oral route as well as your in vivo route, is intravenous route. So these are the most common routes that we do. Other than that, we sometimes do different type of discrete as well as cassette PK that I will later on I will discuss what is discrete PK and what is cassette PK. Then we do group study as well as group PK studies definitely. Then parallel and crossover design, multiple routes of administration, and definitely multiple matrices for analysis like collection of plasma, fishes, urine. So that all these things you just need to know what are the drug concentration in those matrices and how it affects your pharmacokinetic property of the molecule. And then definitely, if your target is CNS, means your drug will act in the CNS, means central nervous system, then definitely it has to cross the blood-brain barrier. So for that kind of setup, as your molecule is designed to cross the blood-brain barrier, so you need to set up an experiment so that that data of that particular experiment will say you whether that molecule will pass the blood-brain barrier or not. So that after that, then those molecules you can test in animal model and you can collect the brain of that animal and you can see the concentration of the drug in the blood, whether that reaches to the brain or not. At the same time, you can do simultaneous PK study as well as you can take the sample and from that sample you can do some biomarker analysis because you may have some target to elevate some biomarker. So you are administering the drug and you are getting the profile in different time course, and then in different time course if you analyze the biomarker level whether it's increasing or decreasing as per your therapeutic target, whether that will be elevated or it will be decreased, you can quantify that also. Now you just different type of PK approach. I would say a different type of PK approach mostly you do in industry, that is that discrete PK that already I told you, that conventional PK, and then cassette PK definitely, cassette dosing PK, and then cassette accelerator rapid rate PK, that is CAST, and then snapshot PK. So these are the 4 studies basically we do in industry. Each and every experiment has different type of setup with different type of advantages and disadvantages. Okay, so I will start from snapshot PK. So if you think about the snapshot PK that you can do in mouse and rat, that already I told you, always a preclinical species we try to do the experiment in vivo experiment in smaller species. So here that number of animal is low, is 2, n equal to 2, number of animals 2 per route, and each animal we used to collect 4 time points only, so maybe 0.5 hour, 1 hour, 2 and 5th hour. Because This is a shorter time span. Up to 5th hour you can take the sample, and from each rat or mice, I would say, you can take 4 sample as well as 2 animals. So here advantage is that you can save a lot of animals because you are doing the 2 animal per compound studies and application of rat or mouse and this, and whatever the parameters you will get from this snapshot PK, that will give you an idea whether that molecule permeates to the GIT membrane or not, and what is the exposure, how much it is, whether it's too low or it's too high. That idea you can get, and it will give you the partial PK parameters. Again, I am telling you partial PK parameter, not a full-fledged PK parameters. This pharmacokinetic parameters. Later I will show you what are the PK parameters that we need to evaluate during our pharmacokinetic studies. Then you think about the cassette accelerated rapid rat PK. So here what we do here actually again number of animal 2 per route, and here time course is little bit higher, up to 6 hours, and then 2 animals per compound, and the sample volume is little bit higher. You can see it's 100 microliter. Here also you can save animal, but snapshot PK and cassette accelerated rapid dosing, basic difference is that in cassette dosing we do multiple compounds, say maybe 3 or 4 or maybe 5 compounds. You are preparing the formulation, there are 5 compounds are there, and that you are dosing that compound with animals. So at a time, in a single shot, you can screen 5 compounds. Can you imagine how much animal you can save, how much time, how much effort, how much money you can save with this study? Again, it has some drawback, definitely. Application to rat only, and the PK parameters, what you will get, that will be the partial PK parameters. And an idea, again I am telling you, it will give you an idea. Whenever you have nothing, means you have nothing about in vivo data of that particular new chemical entity. Whatever data you have, all are basically ADME, means in vitro data. So from that, if you want to explore the amount or concentration of the drug in vivo, how much it will be, how it will be cleared from the body. So in that case, you will get an idea from the cassette car study. And then about cassette dosing. Cassette dosing again, the cassette accelerated and cassette dosing, basic difference is the number of animal in cassette dosing is more, and over a time course of maybe more than 24 hours, sometimes it's more than 24, maybe 48 hours also. So basic difference is that in cars, actually you have up to 6 hours, over the 6 hours, here you have the time over 24 hours. And here time points is 6 to 8 animals per compound is for IV n equal to 3, and for PO is for allute is n equal to 3. And here number of time points per animal is 6 to 12. Most of the case it becomes 6 to 9. So you can see that here if you have 6 to 9 time points over a 24-hour time course, and you have the concentration over time, there you will get the data of 5 compounds, full PK parameters. Again I am telling you, you will give a full PK parameters, but problem is that only you are combining 5 new chemical entities at a time together. So as there are 5 compounds, so data whatever you are getting, so PK parameters you are getting for 5 compounds. There also you are saving time, effort, money as well as animals. But problem is that as there are 5 compounds, so one compound may inhibit some enzymes that will be detrimental for the other compound. So the PK parameters, whatever you are getting, that may not actually reflect your actual PK parameters. When you will be doing the PK study with a single compound, there you will get the actual data or actual PK parameters of the compound. Now you think about the full PK mouse or rat. Here, one compound per animal. So here, the cassette PK, there are 5 compounds in an animal. Here, 1 compound per animal. So maybe 3 compound for oral and 3 animal for oral route as well as 3 animal for intravenous route. So whatever PK parameters you will get for a particular compound, that is absolutely reflect the PK parameter of that particular new chemical entity. So this is a full-fledged An advantage is that you will get the full PK parameters. What are the disadvantages of other three? Here you will get the full PK parameters of a particular compound, no drug-drug interaction, what you used to get for cassette dosing, means N-in-1 dosing, multiple compounds in a single formulation, and that formulation you are administering to the animal. And disadvantage is that labor intensive as well as not cost effective definitely, because for 5 compounds you need to do 5 studies. So it's definitely cost effective, no, definitely it will not be cost effective. Now it is a little bit more elaborated version of cassette PK and discrete PK, because these two are mostly we do in industry, cassette dosing PK as well as a single full-fledged PK. So just cassette PK as well as single dose PK, just see the advantage and disadvantage that already I have discussed. So in cassette dosing or conventional dosing, what we do that that analysis of the 5 compounds, and you are generating a sample of say 1 plate for single cassette PK, and there are 5 molecules are there, and you can analyze the samples and you can get the report for 5 compounds at a time. And for conventional dosing or for single animal single compound dosing, there you will get only the PK data of 1 compound. Definitely low throughput, definitely you can see that low throughput. I already have told you that single dosing, single molecule and single animal, that will give you less throughput. Not cost effective that much. And now that this is a typical study design of intravenous as well as oral route. So here if you see that a different type of rat and mice we used to do here. So the product that we used to maintain here 0.2 mg per kg single dose and 5 ml per kg for without anesthesia and 10 mg per kg for single dose. So this basically a typical study design. What we do whenever whenever new molecule or new chemical entity comes to the laboratory for animal testing, you don't have anything. Just you have some in vitro data. So you need to do some experiments. So what you will do, whether you will do for higher dose, lower dose, you don't have any idea. So in that case, we try to do say maybe 1 mg or 2 mg per kg for intravenous route, because I don't have anything data about that molecule. So we try to do it 1 mg or 2 mg per kg for IV, and sometimes 5 or 10 mg per kg for oral route. So this is the PK profile for individual, how it looks like, and then intravenous, and then the mean profile of that particular compounds, how it looks like. Now the different PK parameters that I said, different type of PK parameters that we need to calculate to get an idea about the pharmacokinetic features or pharmacokinetic properties of that particular compound. That we know that AUC 0 to T means AUC area under the curve from 0 to T minute, means up to 24 hours we are doing the study, so 0 to T, and then T to infinity, that is AUC T to infinity, and if we combine together we will get from AUC 0 to infinity. So how to calculate those? We have a different type of software to calculate those. Even in Excel based formulation also we can put formulation in Excel, and there we just put the concentration, you can get the different type of parameters. So these different parameters have different values or different I would say impact on the pharmacokinetic properties of the compound. And AUC 0 to T, 0 to infinity, T to infinity, all these actually define the total exposure of the molecule in systemic circulation. And then Tmax, Cmax, that already as a pharmacy professionals we know the Tmax and Cmax, what is the impact. Tmax is the maximum concentration, and Cmax, Tmax they can have the time to achieve that Cmax concentration. And elimination phase, if we see that 0 to that graph, it's an absorption lag phase. So if it's lengthy for a particular molecule, that is not good for that particular molecule, because it should not be so lengthy. Whenever you are preparing a formulation, that lag time you can decrease by formulating your new chemical entities in different type of formulations. Maybe CMC, this carboxyl cellulose, maybe MC2 in combination, maybe PEG or DMSO combination, PEG, soluble, different type of vehicle combinations you can use that to reduce that lag time. And this is a typical type of formulation that we use, zero order formulation, toxic level as well as minimum effective level. So you can see that concentrations which is reaching to the minimum effective level for that particular formulation or that particular new chemical entity will get that activity. If it's beyond to the toxic level, definitely it will show some toxicity. So all these parameters you can explore in non-clinical phase just to have an idea of the pharmacokinetic properties of that particular molecule, and so that based on those PK parameters you can rank order the molecules. So this is my top priority molecule, this is my second top, this is my third top, like that. So whenever that molecule is go to the later stage for developmental stage as well as for clinical trial, there you can sequentially you can push your molecules into this is my top priority molecule, so I will take this molecule first for the clinical trial, definitely for the second, third, like that. So all these pharmacokinetic properties will help you to rank the molecules or the compounds for further development and for clinical trial. Now if you think about that different type of PK parameters, earlier I told clearance, half-life, volume of distribution. So the volume of distribution has one clear-cut impact that I would say. You say you are taking the molecule, that molecule is not eliminating from the body, it's eliminating from the body very slowly. It happened now that you are calculating the volume of distribution of that molecule particular. In pharmacokinetic evaluation you have seen that it has huge volume of distribution. That means where that molecule, because huge, why I am telling, because we have our systemic circulation volume as well as interstitial fluid as well within the cell. So it has some volume. If it goes beyond that, which is volume of distribution itself is saying that it has been distributed to somewhere else, not within the circulation or not within the between the cells fluid or within the cells fluid. It goes beyond that. So in that case, what happens that if that kind of warning you can get in the PK data. If its volume of distribution too high, then you can explore where that molecule goes. Your drug is not eliminating from the body, or it's eliminating from the body very slowly. So there you need to think about where my molecule goes actually. Then you will just for a particular project, I will say you there they have taken the fat tissue of that animal, and there we have seen that lot of drugs are depositing in the adipose tissue. The concentration of tissue particular in adipose tissue is much more than the systemic circulation. Means whatever the concentration you are getting in plasma, that is very less than the concentration you are getting in the tissue. So that is one of the factor that volume of distribution has, how much impact it is. Whenever drug deposits the adipose tissue, that creates a lot of toxicity and adverse effect. It will not be eliminated from the body, it will remain long term within the body, and that will show different type of adverse effect and toxicity. So that is the volume of distribution, and whenever we are studying whether the molecule is highly cleared from the body, whether the molecule is eliminating from the body too fast, or it's retaining in the body for moderate time. Its half-life is maybe 3 hour or 2 hour or maybe 1 hour. So that half-life define you how often you need to take that medicine. Whether it will be taken twice in a day or thrice in a day, that you can decide based on the half-life of that particular molecule. And whenever you are trying to establish the clearance of the molecule, you need to consider total clearance of the molecule, not through the hepatic clearance only. The drug cleared through the liver's metabolism, that is called hepatic, just in a gross I would say that is a hepatic clearance. And if it's eliminating through liver as well as other organs are involved there, like kidney, hepatic, because in kidney there are a lot of cytochrome P450 there, even in intestine also there are a lot of cytochrome P450 esterase, different type of enzymes are there. Whether they are responsible for elimination of the drug, if they are responsible, then we need to calculate that also. Means we need to calculate the total clearance of the molecule from the body, hepatic clearance as well as any other clearance. And then that metabolic clearance that already I told you. Then what is the impact? That already I told you, that half-life that defines the dose regimen, how often you need to take, twice in a day, thrice in a day, it will be like this. And oral viability, that you know, oral viability, that in a gross I would say that rate and extent of drug absorption, means how much quantity of drug reaching to the systemic circulation in a gross, that I would say as a viability. So if a drug is by let's say more than 40% or more than 50%, that is good. But that also depends on the projects. If any project that the molecule are too potent, which is activity say maybe IC50 is maybe 2 nanomolar or like 5 nanomolar. So in that case, if your compound is low bioavailable, maybe 10%, but that is also accepted for that particular series of compound or for that particular program. Why? Because if it reaches to the system saturation maybe 20 nanogram per ml or maybe 50 nanogram per ml, that is quite good or quite enough to get the activity, because IC50 of that particular molecule is very low. But I will get that activity say maybe around 2 nanomolar or 5 nanomolar. Say if it's around 20 or 40, 50 nanomolar, that is quite good enough to get the activity. So in that particular project, low viable compound is quite good for. But in that project, if that molecule having that higher viability, that will be not good, because or it's good but you need to reduce the dose also. So there you need to play with the viability as well as dose and how often you need to take. Now that viability, how that viability you use to calculate? Say, already I told you that whenever you are doing a standard pharmacokinetic study in rodents, so there we do in vivo study as well as in vitro in vivo study in different doses, means maybe in intravenous as well as oral. If you want to calculate the percentage viability through oral route, then you need to administer the drug through oral route, and at the same time you need to administer the drug through the intravenous route. Those may not be same, may be same. It's up to you. So whenever you are calculating the viability, so the viability you are getting through the oral route, that mostly we used to calculate through AUC 0 to infinity, that you are getting through the oral route, divided by dose of that oral route. So you are using 10 mg per kg or maybe 5 mg per kg. So AUC divided by dose of oral route divided by AUC of intravenous divided by AUC dose, dose of the intravenous route. So if it's 1 mg per kg through intravenous, then AUC divided by 1, and for the denominator it will be AUC PO divided by dose. I mean dose normalized AUC of oral divided by dose normalized AUC of intravenous into 100. That is your absolute viability, and that you can calculate through subcutaneous route also. But always you have the data of AUC from intravenous route. Now just think about the different type of that earlier I told you, that absorption, distribution and elimination. So different phases are there. So different phases has different role, and we know that activity of a particular drug that depends on how much free it is inside the circulation. If a drug is 90% bound, means 10% free, that 10% is available to bind with your receptor, and that will be available for your metabolism, and that is available for to inhibit the enzyme. So all these are depends on the percentage binding as well as free. And at the same time you also need to define whether drug is eliminating from the body after metabolism or drug itself is eliminating from the body as unchanged. That also you need to establish in pharmacokinetic phases. So if all those parameters we have in our non-clinical stages, I mean before going to clinical trial, if you have all those parameters and all those properties, pharmacokinetic properties of that particular compound, then definitely you can reduce the attrition rate that already I mentioned to you, that is close to 40% of the drug fails in clinical trials just because of the poor pharmacokinetic properties of a particular series of compound. So if you have all those parameters and you know for that particular program, I am trying to get this much of AUC, this much of Cmax, and your Tmax should be this much, and your total viability it should be not less than 10%, it should not be not more than 90%, or it should not be less than 40%. So that particular design, everything parameters and things parameters and data you have in the clinical preclinical stage before going to clinical trial. And there are different parameters are there to that affects basically viability. I think as a pharmaceutical person that most of us are aware about that. That drug related factors, that solubility, stability, permeability and particle size. These are the factors that affect the viability. Similarly, formulation factors are there. That any molecules which is not solubilizing in the GIT fluid, that will give you less viability. And similarly, physiochemical factors that is gastric pH. If your molecule is unstable in gastric pH, means acidic pH, then you need to deliver that particular molecule for maybe in intestinal pH, or if it not stable intestinal pH, you can, because stomach and intestine, these are the region most of the drugs absorb in that particular window. So any drug which is going to the systemic circulation, that has to face this region, in stomach to intestine, and the pH from 1.2 to around 7, 6, 6.8 like that. Not beyond colon like that, because there are pH is more than 7 or 8. So different type of transporter proteins are there. Transporter proteins has two roles, I would say. One is it helps to penetrate the drug from GIT membrane to the cell or maybe to the circulation, and there are another proteins that will eliminate the drug from the drug enter into the cell, and there is a transporter that will influx from the cell to the GIT circulation again. So that is the influx protein. So whenever you are trying to establish a molecule, whether that's a PGP substrate, means influx protein substrate or not, that also you need to establish. If it's influx protein substrate, means it is getting out of the cell, then how much it is, how extent it is, that you need to establish. So these are the species that earlier I told you, that from smaller species to higher species, finally is clinical trial, and then definitely different type of. So during preclinical phase, actually we try to establish not only pharmacokinetic properties, we also try to establish toxicity as well as the other parameters, different physiochemical parameters, different type of different viability, different animals, different type of metabolites across species. Whether say I am getting 3 metabolites for a particular species, maybe in rat, whether those 3 metabolites are forming in dog or in monkey or not. That also you need to establish. If it's not, if it's different, if there are any unique metabolite which is forming in cynomolgus monkey, then you need to specially screen that particular metabolite, whether that, because that's a peculiar metabolite that may show adverse effect in human trial. So that also you need to consider. So whenever you are doing the metabolite profiling, you need to establish that profiling of the metabolite across species, so that you can say that there is non-peculiar metabolite that is forming in dog or maybe in mice or maybe in monkey. If there are any peculiar metabolite, that you need to consider or you need to validate that. So now that you know all the parameters or pharmacokinetic properties of that particular compound, and now you are going to the clinical trial. Now you are safe, because you are investing a lot of money for the clinical trial, because when you are doing to the clinical trial, if your molecule has any toxicity or adverse effect due to that, in phase 1, the clinical trial healthy volunteer, if anyone dies, then it's your responsibility and you have to put a lot of money as a compensation for that particular death. So how much burden it is, let's say you can imagine how much burden it is as a cost burden. You are investing a lot of money in drug discovery, and in later stage when you are going to the clinical trial, there you have seen that your molecule has so much of toxicity and your healthy volunteer in the phase 1 is right. So in that case, to avoid that case actually, is to avoid that any unwanted adverse effect in clinical trial, we need to establish in that preclinical phase, and that we can do through pharmacokinetic studies as well as different type of toxicology studies. And definitely we can reduce the attrition rate. That only I have told you that 40% of the drug was failed due to poor pharmacokinetic properties. Now you have all the properties, you can establish, okay, I have this much of molecule, I have ranked based on the PK parameter, I should take this molecule first and then this molecule second. So based on that, you can say, because whatever the investment you will do in the clinical trial, that is that will give you I would say return that will help you to get to have an understanding whether that molecule will be clicked for the next level or not, or it will be further developed or not. And definitely it will help that medicinal chemist that earlier I told you to modify the molecule, whether the molecule has soft spot and efficacy site is same, and then binding site same. Accordingly, you can design the molecule and you can further develop that particular project. And a medical chemist can design the molecule in a better way. And definitely, if a particular site of the say the molecule is metabolizing at a particular site, and because of that you are getting a toxic metabolite. And if you modify that particular site, metabolism not happening in that particular site, so that molecule, whatever the method is generating now, that is set. So that also you can get an idea in this page. So, so this is all about. Thank you for your patience, for listening. Please, if you have any clarification or question, please, I will ask you. Please.
Thank you.
Good afternoon, sir.
Yes.
Sir, actually, I wanted to ask why this cross-species metabolic profiling is necessary. The first question, and the second question is how to determine the soft spots in a molecule.
Thank you for a nice question. So, shall I go back to the slide? First, I would say that why I need to do metabolic profiling across species. So, you are trying to say you have a lead chemical entity that already told you the lead chemical entity means you don't have any information about the structural modification of that particular molecule in presence of metabolic enzyme, maybe liver enzyme, liver microsome, maybe hepatocytes, or different type of enzyme. So, say I am doing metabolic profiling in rat, and there I receive 4 metabolites, and for the same molecule, rat means rat liver microsome. Okay, because that is the in vitro. Because initially, whenever we do metabolic profiling, we try to do it in vitro assay first. Means not from in vivo animal. In vitro, so we are incubating the compounds and liver microsome. What are the metabolites or structural modification is happening in presence of liver microsome, that you need to establish. And then, so you are getting 3 or 4 metabolites. Then you go to the dog, legal dog, liver microsome, you are incubating, and you are getting the number of metabolites. If it's same metabolites, then there is no issue. If there is any peculiar metabolite, say you are getting 4 metabolites, then in structural modification, you have 1, 2, 3. Yes. Say if you just consider this case, okay? Here, if you think that the parent molecule itself, the top is a parent molecule, you can see the structure. There are 3 segments that I have highlighted with the red. So those are the 3 soft spots. That modification is happening in those particular 3 spots. Okay? So that S double bond NH, that you can say that is converting to S double bond O, and 1 methane is there, it's there. That NH particular is leaving from the sulfur atom. So, and whether that particular that metabolic 5 M5 is happening for all other species or not. If any peculiar metabolism or peculiar metabolite is forming any other species, that you need to consider before going to clinical trial. Because whenever you are going to clinical trial, you don't have any information, in vivo information, but definitely in laboratory, in vitro experiment you can establish, because we have liver microsome from rat, dog, monkey, as well as from human also. Because there are liver microsome, pooled human liver microsome, that mostly generated from liver definitely. Maybe person who is dying accidentally or something, kind of that mostly donated liver basically. Yes, mostly donated liver. So from that donated liver we used to collect liver microsome, and that liver microsome you can put into the reaction mixture in vitro assay, and there you can see the number of metabolites forming in human liver microsome, and that you can compare with the rat, dog, or monkey. If it's different type, it's whatever the metabolic pattern you are getting in human, and that is similar to rat or dog, that is fine. But if it's different, then definitely you need to consider it. And if you characterize the structure of particular peculiar metabolite, I would say peculiar metabolite, then definitely in laboratory you need to synthesize that particular metabolite, and you need to profiling that. Profiling in the sense whether that has any adverse effect or not, that you need to establish. So, yeah. Yeah. No, I am not stopping here. So, say molecule in human liver microsome, I am getting one peculiar metabolite.
But we don't know what it is going to be before clinical trial.
That is fine. That is fine because that's why I'm telling. Whenever you going for in vivo study, you are getting data from in vitro, and that is human liver microsome. It's not human in vivo condition. It's human liver microsome because it is assumed that most of the drugs are metabolized in the liver. So whatever the metabolites we will get in liver microsomal, human liver microsomal in vitro, that is expected to be there in vivo. That is extra protein value. It's not exactly what we are getting in vitro human liver microsomal. Yes, it's probability. Because I don't have data in clinical trial, no? So before going to that, I must have some data. So that's the data in vitro liver microsome I am getting.
But one need to establish the potential harm of that metabolite.
If in vitro you are getting any idea, then definitely you have to establish.
You have to establish.
Yes, nice. And for establishing that, you need to chemically synthesize that particular metabolite in laboratory. Because new chemical entity, whenever we are synthesizing in lab, so if I have the structure of that particular metabolite, say NMR data, mass spec data, iron data, so everything data I have, I know the structure of that particular metabolite, and in chemical laboratory we are synthesizing that molecule, and we are establishing the safety, efficacy, if there anything for that particular metabolite.
Once again you have to reroute.
Yes, reboot that for that particular metabolite only. Because everything I have for that particular metabolite I need to follow all those parameters. Maybe we need to first establish the safety profile of that particular metabolite if it's there. Second question is whether how we can identify that particular soft spot. That's a nice question, I would say. So LCMS one instrument is there. I think all of us know LCMS. So in LCMS, what we do? What I told you that molecule has been incubated in liver microsome system. So there number of metals are forming, and you are getting the samples from that particular assay. And from there you have the structure, and from the parent compound itself, you need to fragment. Say I am the parent molecule, and I have mass around, say it's 409. So my molecular weight is at 408, and in mass, in positive polarity, because it has two polarity, positive and negative. If it's ionized in that molecule in positive polarity, it will be one unit will be added. This one hydrogen will be added with the anion, so it will get 409. So whenever, say my ion, my mass ion M by Z, the parent ion is 409. When I enter into the mass, the mass will fragment me in different segments, say maybe left hand, right hand, finger, toes, legs and everything. And each and everything has different type of mass. Say one finger maybe say 2 gram, just for example, say 200. So one left hand is 350 gram. One leg is say 280. Other leg is say around 270. So all these different type of fragments I will get in the mass. So all different fragments will come from the different segment of the molecule. So it's a big structure. So this part is coming 200, this part is coming 250, this coming 220, this part is coming 180. So that expertise you must have to identify the ions, that ions are coming from the particular part of that molecule. Okay? So you have that fragment or ions, and from that ion, whenever you are getting the metabolite and you are fragmenting that metabolite, and say this part is 200 and you are 200 plus 60, means 200 to 116. That means, say left hand side, one modification is there, that is 60 unit added in the left hand side. So this is a structural features whole, and in this part some modification is happening, some oxidation is happening. So in left hand side I am getting oxidation, this plus OH. So that is the metabolic short spot, but it's a bit tricky and definitely challenging, and definitely you should have that much of expertise to get that. It's not a single day experiment, you are doing this and tomorrow you are getting the data. It's a labor intensive as well, and definitely you need some skill setup as well as expertise also, because you need to have some idea about the fragmentation pattern of the molecule, from which part of that molecule is coming, and accordingly you just interpret. Not chemist, as a biologist, in fact I also do that, it's not a big issue. Definitely when you have that expertise, when you are working a lot with the mass spec as well as the structure, you will come to know that based on the structure you can say this part will be modified. Means that say you are doing 100 to 100 compounds and across species, maybe 2 or 3 species, and you will come to know after just seeing the structure, you can predict that this could be this is the position maybe modified. This could be the position maybe modified. That Based on the structure, you can guess. And definitely, yes, definitely you can guess. Yeah, definitely. Definitely you get some idea. And then you just see the experimental data. Whether your assumption is right or wrong. It's not 100% accurate. I am telling you, it's not 100% accurate. What you are guessing from the just seeing the structure, that may not 100% reflect in your experimental data. That you need to interpret based on the fragmentation pattern of the compound as well as the metabolite.
Okay, thank you.
Please, any?
Thank you, Dr. Bera, for your lucid, in-depth discussion on the particular topic, which is a very fundamental topic, and each and every pharma professional should understand and remember it. So you have covered most of the relevant subtopic, which include steps of drug discovery phases, importance of pharmacokinetic study in drug discovery, importance of ADME, metabolism, particularly clearance and VD, apparent volume of distribution, and its influence on drug discovery process. These all the fundamental things each and everybody should know and understand. So I congratulate him for his beautiful lucid deliberations. Whether we'll go for invite any question?
Can you throw some light on low intrinsic clearance drug?
Low intrinsic clearance drug, antisalinic acid is one of the low clearance molecule. Means it will be eliminated from the body very slowly. And one more thing, that most of the digoxin probably has a low clearance. Digoxin also low clearance molecule. And that low clearance molecule that happen mostly, it will not be eliminated from the body. I think you know that. Digoxin will also be low clearance. I think Dr. Nandi can help me, please.
A low clearance molecule categorized based on the unit protein.
Yes.
On the basis of extraction ratio, you can say hepatic extraction ratio. If it is a 0.3 or less, then it can be categorized as 0.3. If it is 0.3 to 0.7, it is moderate, and about 0.7 is high. Based on the in vitro metabolistic data, two important parameters you get. One is intrinsic clearance or corresponding theta, and if you include the hepatic clearance into the behavior, you have what the blood how much of body weight of the animal, body weight to organ to body weight to the liver, then you will get the hepatic extraction. This means how much go, how much out. In case of low extraction ratio, diazepam simply low goes to the in vitro study. Diazepam is low so low, you have to address the low clearance. So you are inquiring about the low clearance molecule. Diazepam and alcohol is the best right? Because it is the only issue of that molecule, that's why people are comfortable to take, because in the earlier molecule, there is liquidity of the molecule. Right now, modify the benzodiazepine structure to alpha-zolam, that's very common.
Even just I want to add one more thing. A molecule maybe have low clearance in rat that may not be reflected in human also. It may be high clear in human. So again, it's a kind of, you need to establish that in across species. Whenever you are doing metabolic stability across species, there you can establish whether that molecule is, because final target is to establish in human, in clinical trial. So the drug is whether low clear in human liver microbiome also, that also you need to consider. Sometimes it happens that it's rat, it's low clear, but in human liver microbiome it's moderate to low, sometimes it happens. This across species may change.
Thank you, Dr. Bera. I would like to request the organizer to proceed for further.
Thank you.
Thank you so much, sir, for sharing such an valuable information and thought-provoking session. I now request our chairperson, Dr. Majumdar, to present a token of gratitude to our resource person, Dr. Rammohan Bera. Thank you, sir. Now I request Dr. Saurabh Ghosh to kindly felicitate our Honorable Chairperson, Dr. Shubhobroto Majumdar. And that concludes session 5. Now we will move on to our last scientific lecture session. I request our respected chairperson, Shoili Choudhury, Professor and HOD of the Department and Pharmaceutical Chemistry, BCDA, College of Pharmacy and Technology, Kolkata, along with our distinguished resource person, Mr. Deborshi Dattagupta, the Managing Director of East India Pharmaceutical Works Limited, to kindly come up on stage and take their seats. We are privileged to have with us Dr. Shoili Choudhury, Professor and HOD of Department of Pharmaceutical Chemistry, BCDA, College of Pharmacy and Technology, Kolkata, as chairperson for this session. Dr. Choudhury is an established researcher and academic in pharmaceutical chemistry. She has made significant contributions in pharmaceutical sciences through her research, publications, and academic leadership. Her research spans pharmaceutical chemistry, phytochemical studies, and International Journal of Pharmacognosy and Phytochemical Research. She has presented widely with 28 papers in national conference proceedings and 3 in international conferences, reflecting her active engagement in scientific community. Beyond research, Dr. Choudhury has contributed to organizing scientific conferences and promoting industry-academia collaborations, strengthening BCDA College's role as a hub of innovation and knowledge sharing. Her consistent academic output highlights her commitment to advancing phytochemistry, pharmaceutical technology, and innovative approaches to pharmacy education, making her a valued scholar and educator in the field. May I now request Dr. Shoili Choudhury to kindly introduce our resource person, Mr. Deborshi Dattagupta, and would request to extend up to the question-answer segment of this session. Please, ma'am. Hello.
First of all, I would like to extend my heartfelt thanks to Adamas University for the invitation and to session the chair for our today's honorable speaker, Mr. Debashri Dattagupta. Mr. Debashri Dattagupta is the Managing Director of East India Pharmaceutical Works Limited, a Kolkata-based company with profound legacy dating back to 1936 with a vision of providing affordable, high-quality medicines under the motto, Medicine to Millions. An alumnus of Calcutta University, Department of Law, Mr. Dattagupta began his professional career as an advocate at Calcutta High Court in the year 2001, before taking on leadership in East India Pharmaceutical Works Limited in the year 2003. A law graduate, he represents the third generation of family legacy of nation-focused pharmaceutical entrepreneurship, being the grandson of freedom fighter and former EIPW Managing Director Hirendranath Dattagupta. Under his stewardship, EIPW has strengthened his commitment to providing affordable, high-quality medicines under its guiding motto, Medicine for Millionaire. His leadership blends legal modern challenges while staying true to its founding principles. Beyond his role at East India Pharmaceutical Works Limited, Mr. Dattagupta has held numerous distinguished positions, including the Vice Chairman of the Indian Institute of Packaging, Government of India, Vice Chairman of the National Safety Council, West Bengal Chapter, Vice President of State Productivity Council, West Bengal, Chairman of the Eastern Region of the Indian Chemical Council, that is ICC, and he has also served as Vice President East of the Ayurvedic Drug Manufacturing Association and as an executive committee member of the Bengal Chamber of Commerce and Industry. So, respected for his vision and leadership with the reappointment as, again, Managing Director of East India Pharmaceutical Works Limited in September 2024, reaffirming the Board of Confidence and his ability to guide the organization forward. So we are happy and as well as we are honored to have you, sir. Just a small request, I will request kindly to adhere with the time limit of 30 minutes as we are running somewhat late to ensure the smooth conduction of the proceeding further. Thank you, sir. Now the stage is yours.
Thank you, ma'am. Yes, I am aware that this program is running off almost 1.5 hours late, so I'll keep my session down to 20 minutes. I'll just ask two questions. So I can see two kind of faces. So don't worry, 15 to 20 minutes is all I will take. First of all, I'm happy to be here. It's quite a distance from where I stay. Number two is, I don't think it's a scientific session. I will just try and present my views on the industry perspectives because I am part of the manufacturing industry. So whatever you have heard throughout the morning is how to invent a drug, right? So my responsibility is how to make a drug and how to sell it. Right? Even if you invent, if you do not make the drug or you can sell to the customers at large, I think everybody, all scientists will look forward to inventing something that helps society at large. So in pharmaceuticals or drug development, the end point is the product, and that is where I come in. And I'm sure many of you will spend a lot more time in the industry in the coming future. Be it as entrepreneurs, of course, I would love everybody in this room to become entrepreneurs, or part of the industry. But in any way, you will contribute to the society as large in providing pharmaceutical or healthcare solutions in the future. But as we are actually talking about industry and academia partnership, I think it's a love-hate relationship with the manufacturing industries. we keep on. The time I joined the industry in 2003, I've been in innumerable sessions where we have discussed how to develop or increase the industry-academia partnership. Okay. So I'll be very frank here. I mean, there's nothing to be diplomatic. The industry always think যে ওরা খালি পড়াশোনা করে আর পেপার লেখে, and the academic thinks ওরা খালি বেচতে জানে আর পয়সা করতে জানে. This is a very simple term what has happened. But the fact is, if both of us are not there, the end product does not reach the customer, right? Ultimately, what is important is the customer should get a solution in healthcare which helps him or her cure their disease, right? That's the end point. This is a big thing. I think the scientists who have been spoken, I was hearing, very technical though, as I am from the legal side, goes to 100 kilometers above my head, most of it. But the fact is that the industry, by the way, how many of you judge industry by seeing Shark Tank? Have you seen Shark Tank? Very popular program, right? That's not the industry, unfortunately. That gives you good TRP on TV though. Looks good on TV. When you see drama in the studio, right? When somebody pitches some idea and the sharks are trying to negotiate a deal for both of us. That is actually that's not how industry works in the real world. It is a little bit similar, but the industry or the manufacturing industry which I represent, or I would say the MSME manufacturing industry of pharmaceuticals which I represent, it is a little bit different. As you heard from the introduction, I come from a 90-year-old company, a company formed by freedom fighters. So they had a different, this of course it's a commercial organization. We are not here for charity, and neither are you. When you join the industry, when you join your profession, you will expect to get paid, right? It's not a free thing. It's not a free thing. So this is a commercial organization, but we have always followed a vision where we have thought of having high quality medicines, and it goes to everybody. Nowadays, of course, you kept on hearing high quality medicines at an affordable price, high quality medicines at an affordable price. Nowadays, the notions are a little bit different, because you can see everything is getting expensive, and so is medicines. Manufacturing of medicines are under very strict regulations, very strict. As you are in this field of study, Schedule M of Drugs and Cosmetics Act, or the CGMP, the drug policy gets updated every year. Recently, the new drug policy has come, which leads to very, very strict processes of making medicines. Okay. So, when we again going back to the relationship, right? There are always a lot of negativity, but good things have also happened. I brought a small list so that I can just let you know that when industry and academia has joined hands, what has happened. I'll just give you a few examples of few products. Okay. Centroman is a contraceptive pill, developed by the Central Drug Research Institute, or CDRI, Lucknow, and manufactured by Torrent. Okay. That is how you develop, and the drug comes to the market, so that people can take advantage of it. Right? One example. Another one, CSIR and Intech Chandigarh, they collaborated with Nostrum Pharmaceuticals of USA to develop the crop-specific streptokinase CSSK, again another product that came into the market. When you come to our organization, we did a project with CSIR 11 years back, where we did anti-prostate inflammatory drug. Okay, that is a proprietary Ayurvedic drug, comes under Ayush now. So Prostalin was developed with CSIR and IICD in Kolkata, and in our labs as well, in our company, and the product is in the market. So that is what happens when things do work out. But there are very, very few examples of this happening, which is very alarming. Very alarming. So what we need to do is to come down from all of our preconceived notions and start joining hands. Because if that happens, the society gets products which the society needs. Otherwise, if you see every year, thousands of patents are filed in India. Thousands of patents. And how many of it is granted and how many of it comes as a proper product? It's very difficult. Developing a drug is absolutely difficult. Takes years and crores of rupees to develop. So for different organizations, I think the gentleman who was presenting before me, he was an ex-Dr. Reddy's, if I'm not, that is what I heard. So he knows the limitations of research in a manufacturing organization. Okay? So we all also have our research labs. So for us as an organization, we have to do three things. We have to not necessarily invent, manufacture and sell. So all these three things are very, very important, but big areas in itself. Okay? Drug development itself is a huge area of work. Drug manufacturing is a huge area of work. I hope Adamus sends few people to our plant to see what happens when drug is manufactured according to the best practices in Indian law. And selling of a drug. That's a very, very different, difficult area. So for all of you, you will be coming into the industry in one of these three areas in the future, right? Either you will invent, or you will make, or you will sell. There's nothing outside. But let me tell you that the drug market in India, which you also should know, so that you can prepare for the future, you should know the market where you are coming in the future. India has crossed 1 lakh crore of rupees in the drug market. 50% is exports, 50% is domestic. 10,000 registered pharmaceutical companies already, with around 8,000 MSMEs. So for all of you, there is a huge industry for you to grow. There is a huge market for you to exploit. There are ample number of companies. I'm talking about pharma companies. Apart from that, there are research-based organizations as well, where you can go. Okay. So for that, what is needed is a very strong industry academia partnership, so that you can have a little bit of knowledge of what the industry is all about. It is difficult for you to understand what the industry is sitting here in your classrooms. So the more your university sends you to university, to different industries, the better you will have an idea on what happens there. Otherwise, you will have a wrong notion or a different notion that maybe the industry is not conducive. No. A big example is all of you survived COVID, right? You've survived the toughest situation in the last 100 years and the toughest span of challenge in recorded modern history. All of you have survived, that is why you are here. So can you imagine at that time, the only industry that was left untouched in terms of growth was the healthcare industry. It's an unfortunate event. COVID was an unfortunate event. But the healthcare industry was the one that grew, that never stopped, and contributed to the well-being of the country. You heard of Covaxin, right? Covaxin is also an industry academia partnership product, right? Right? You do know that? Covaxin was a product of industry academia partnership. You know? You did not know. So vaccine did help, right? Vaccine, I think did help. I know I'm not going to open a Pandora's box here. But it did help in one way or the other. So whatever happens, whatever the industry thinks, whatever the academia thinks, the end point is that this is a complementary handshake. Right? We can't do without you. Right? And for you, and your dreams to take shape in terms of an invention or a drug, which comes down to a bottle of liquid or a tablet, you can't do without us as well. Okay? So I think that it is time that after years of speaking, everybody, we still talk about this, right? That how can we unlock the potential of the industry academia partnership? We are still talking about it. I know a lot of things are happening because we do get a lot of people from the academics to our plant for internships. They are very good. Few of them are very good. Okay. So when they come, we understand. When we see them, when we see quality in them, we understand that their teachers must have been doing the right thing with them. And of course, the industry looks forward to people who can, of course, make a difference. Of course, you will be there to build up your career. But the industry will ask for quality when it comes to hiring all of you. I know this is off topic, but I was told that I should mention this to you specifically, that what the industry expects from all of you. The industry will expect hard work and honesty. Apart from that, I don't think the industry expects anything from you. They want hard work, which of course will come from your education and your approach, or what the university teaches you, and honesty. So this is what the industry wants. So I think that you should know that the industry is there waiting for all of you in whatever format. And again, I'm saying this again, I would love all of you to become entrepreneurs yourself. But yes, for that, you need to grind actually yourself in the industry first. Otherwise, you won't understand how to build up your own company as well. In my case, I was lucky because I'm born in a family of industrialists. But yes, I did do my time elsewhere in the legal field because pharma is a highly regulated field. All of you, do you have pharma laws when you study? Do you have laws? Any subject on laws? You have, right? This is highly regulated industry. Okay. So I'm very sure that is going to happen. And for that, I think your university plays a big role. I would love to invite all of you to my plant, including the professors. I always believe, with all due respect, I think the professors, those have very limited industry exposures, should come as well. Because if they know what the industry is all about, it is easier for them as well to understand what the environment is all about. So the industry is should not be misunderstood. Okay. Now I have received multiple comments during COVID, and a few of my apartment residents that no, it's not that. COVID was a terrible time for us as well, because I remember that because being in part of a essential services industry, we could never close down. So even with all this threat, everybody worked. Not in the initial days because it was not possible to travel. But modern pharmaceutical manufacturing plants are very closed areas, very small closed areas with modern machines. You work in a very closed environment. So everybody had a threat of infection. But the industry did what it was supposed to do, provide medicines. There were hardly any shortages of the general medicines which was needed for to fight COVID. So like paracetamol, I believe that it was one of the most used medicine in COVID, apart from the specific ones. So for you, the industry is open. I'm happy that you will be there taking all of our industries forward in a few years. With just one word, everything will work out when your entry into the interview room is full of confidence. I am part of my company's interview panel sometimes, and the only thing I look is the first 5 seconds when somebody walks in. That says a lot about you, the first 5 minutes. Okay. So I'm sure that your professors have done the right thing and have taught you well, made you ready for all of you to have a fantastic career, again, as part of the industry or being the industry yourself. And I will look forward to all of you to see your names as big entrepreneurs in the future, okay, so that you can contribute to the society at large. Again, this is not a charity, so make sure that you do the right thing you invent the right product, you make sure that it comes down as a product. And you have a good handholding with the government and the industry so that the people of our country and people of the world, because as you know, India is the largest exporter of generic medicines in the world. 60% of generic medicines used in the world are from India. So we dominate the field of generic manufacturing in the world, and we should keep dominating. Okay? We are proud as a country that we have done this, and all of you will be contributing to that in some way or the other. So thank you so much. I'm very sorry that I'm cutting it short because again, when you join the industry, you will understand what month end means. Okay, but that is the different thing. So I need to go. But anytime you need anything from our side as an organization, please let us know. You're most welcome to come to our plant, including your teachers. I'm inviting all of you. If you want to know something from me personally, I leave my email address and phone number with your teachers. So thank you so much, and please have a good lunch. I'm sorry that I have kept you waiting. Thank you so much. Jai Hind.
Thank you, Mr. Dattagupta, for the wonderful talk and motivating all the audience to become the entrepreneur, to open up all the probable ways of how we can collaborate with the industry as well as the academia. Thank you for the session. Now I am asking from the audience if any questions are there today. Any question from the audience? I think you people will not be getting such opportunity to interact with him, so. Any more question?
Thank you, sir, for such an insightful presentation. Sorry for asking you while sitting. And sir, actually, my point is, we as students, I'm from pharmacy, Bachelor's of Pharmacy, I'm doing right now. So we are still not pretty much aware that how the market is actually functioning, because we don't have the real-time knowledge, to be honest. And we all are worried about our careers, how to step forward. And many of us do want to pursue higher education. Many of us wants to complete their M.Pharm Masters. So my question is, there are various departments, there are various sectors in industry. So in which pathway there is higher possibility or prospect to grow as a fresher, as a student from my point of view, that how should I pave my pathway and in which direction that will lead into much more higher possibilities so that we can flourish, because we do ask our fellow mates, we do ask our people around us to grasp some knowledge, but it's not always enough, it's always doubtful. So I would like to ask you that.
It's a very wide area of questioning, but I do understand your concerns. It's very natural. If I was in your place, I would have the same concern as well. You see, the pharmaceutical industry is very large. So, as you study different topics in your curriculum, you might have a specific area of interest. Everybody has that, right? For example, when I studied law, I had a lot of interest in constitutional law. Don't ask me why, but it's a very dry subject, but I loved it because my senior was in constitutional law. But I liked it. So you must be having some inclination towards some area of work, maybe research, or you want to go to marketing, or you want to manufacture. Okay. So for you, again, that is where the fact that you should spend some time in the industry, in form of internships, of course, is required. Where you can see a lot of areas actually in a practical way. Because here you are actually studying, right? It's from the book and from experiences of your teachers. But the moment you personally go inside an organization, maybe you are part of a sales team as an intern, or part of the manufacturing team as an intern, you will have a better idea, because whatever you are reading now, it takes shape in a different way in the industry. That's the ground level practical area of work. So I think that, I hope that you can carry on with internships so that you can understand, because there's nothing better than having a hand-to-hand experience in the industry. So I think that's one way out, because same thing happened with me. I had a lot of thoughts in my mind when I finished law. What will I do? Because criminal law is glamorous. Right? You see it in the movies. Everybody thinks everybody is a criminal lawyer. So I decided, I was thinking what to do. And I asked my senior, I said, you need to go to the court and find out what you want to do. Because if you don't go to the court, then you won't understand anything. Of course, it's not the similar comparison, but best way is to get into the industry, because now the internships are part of your curriculum, right? When we were studying law, there were nothing as internships. You used to personally go to seniors and say, I'm okay, even. So it's easy like that. But now it's part of your curriculum. It's set. You have a set program. And the industries they know that the interns are coming and we need to show them different areas of work. So it's best that you get into the industry as an intern as soon as possible. I don't know whether in the future the time frame of internships will increase or not. So somebody coming in for a week, they don't understand. They can't understand anything. And then when it happens to in our organization, I ask them that, do you want the certificate only? Then you can sit in the library and spend your time. If you don't want to learn, it takes a little bit more time. So I just hope that in the future the internships become a little bit longer. I know it's difficult for you to fit everything in the curriculum, but at least a month is required to at least understand that what part of the process you would like. Because all three are different. Invention is one thing, manufacturing is one thing, selling is another thing. Very interesting, all three of them. Although I am not in the first part, I mean the latter two parts. But maybe, I wish you all the best. I don't know, maybe you might land in our plant as well for internships, I don't know. But for all of you, it's better through your college you have I think a department that facilitates internships. So I think they will help in setting up internships. There are, and I'm not talking about industries in Calcutta as well, because the entire country is open for it. Because I heard, I was in that room where it has a cell for international relations where I heard that the university connects with other universities as well and the industries as well. So I am sure that they will do the necessary and you will end up in different pharma companies so that you can find your place of interest. So all the very best.
Thank you, Mr. Gautam once again, and obviously thank to all the audience. So with this, I am handing over the proceeding to the organizer again. Thank you.
Thank you, sir, for such invaluable insights. I now request Dr. Shoili Chaudhary to present a token of gratitude to our resource person, Mr. Deborshi Dattagupta. Thank you, sir. Now I request Professor Dr. Bishwajit Basu to kindly felicitate our Honorable Chairperson, Dr. Shoili Chaudhary. With this, we conclude our all scientific lecture sessions for the day, and it's a pleasure to have you all here today at this gathering. As we take a break to enjoy our lunch, I want to take a moment to thank each of you for your active participation and enthusiasm throughout the conference. And after lunch, we will proceed for our poster presentation session, and I would request all the participants to take all their belongings with them. And we will be back in this auditorium from the 4 p.m. for the valedictory session and other activities for the conference. Thank you all. And for the certificate collection, it will be done in Convention Center from 2 p.m. to 4 p.m. After that, from the 4 p.m. the valedictory session will be here. So thank
you.